tal? Muy buenos días. Sean bienvenidos a este segundo día de actividades que hemos organizado para ustedes en este cuarto simposio internacional sobre garrapatas y las enfermedades que transmiten. Para darles la bienvenida el día de hoy, vamos a tener como moderador al doctor Juan Joel Mosqueda Gualito, el cual va a estar presentando a los presentadores el día de hoy. Antes, quiero aclarar la dinámica del simposio nuevamente para nuestros participantes. Entonces, quiero decirles que el día de hoy las transmisiones van a seguir a través de YouTube y de Facebook. A través de YouTube van a hacer las transmisiones en inglés y a través de Facebook van a hacer las transmisiones en español. Les recordamos que la liga para el registro de asistencia se va a estar publicando 10 minutos antes de terminada cada ponencia. Entonces, la liga solamente tendrá una duración de 10 minutos y después se bajará para que ustedes estén al pendiente de la liga y puedan hacer su registro. Entonces, sin más, les quiero presentar al doctor Juan Joel Mosqueda Gualito, el cual va a estar a cargo de las presentaciones de los ponentes el día de hoy. Buenos días, doctor Mosqueda.
¿Qué tal? Buenos días, doctor Eleazar. Buenos días a todos. Bienvenidos de nuevo al simposio. En el segundo día, hoy las presentaciones van a ser en inglés. Entonces, a partir de ahora voy a hablar en inglés y cualquier duda, como ustedes pueden ver, como lo mencionó el doctor Eleazar, la transmisión en español será en Facebook. So, good morning, everybody. My name is Juan Mosqueda. Today, we are having our first presentation of this morning is by Dr. Andresa Fish. If she is around, yeah. I'm going to present Dr. Andresa. She is veterinarian. She got a bachelor degree in veterinary medicine in the Federal University of Pelotas in the state of Rio Grande do Sul, Brazil. She got a master's degree in biotechnology in the graduate program in biotechnology at the Technological Development Center at the Federal University of Pelotas. And she is currently a postdoctoral fellow at the Ribeirão Preto Nursing School at Sao Paulo University in Sao Paulo in Brazil. In this postdoctoral position, she has been involved in research projects for the development and evaluation of recombinant vaccines against the cattle and dog ticks, and B and T cell protein epitope mapping in tick antigens, and identification and prediction of peptides bound to bovine MHC class II. Dr. Andresa, please go ahead.
Thank you very much. Good morning, everyone. I would like to thank the organizing committee for inviting me for this talk, and all of you that are giving your time to watch my presentation. So as Dr. Mosqueda already told you, I'm a postdoc fellow in the University of São Paulo. I'm work under the supervision of Dr. Beatriz Ferreira, that is the head of our lab here. And we do work in our lab with both ticks, Rhipicephalus microplus and Rhipicephalus sanguineus, in kind of some research areas. Our main research area is the development of vaccines, recombinant vaccines. That is what I'm going to talk to you about today. But we do have research with tick and host interactions and characterization of tick proteins, okay? So if you can see my presentation, if you can, just let me know, okay?
We can see.
Okay. Can you see the second slide already?
Yes.
Okay. So a lot of people already told you about the importance of ticks. I'm going to skip this a little bit. I know you have another talk later that's going to talk a little bit about it. So our work here, I'm going to present to you now, it's about our efforts to develop Rhipicephalus microplus vaccine. And if we think about the several research that work in anti-tick vaccines, there are several strategies working with different organs and tissues to use as vaccine targets to isolate proteins or sequences. And in our lab, we work with the idea and the proteins that come from tick saliva. So we do know that saliva is important for tick feeding. In saliva, we have several proteins and several other non-protein components, but in our lab, we work with the protein components that have several functions, anti-inflammatory functions, metalloprotease, protease functions, fixation functions. And we do know from several papers that if we can block some of these functions, the tick feeding is impaired. So in our lab, we work with the tick saliva as a source of salivary proteins that will be used as vaccine targets. In our main idea is that if we can neutralize the salivary functions, we can impair tick feeding. And we do like the idea to work with tick saliva because saliva proteins are exposed antigens. So they are presented to the host immune system during the tick feeding, and it can act as a natural boost. So we could probably immunize the animals less times compared to other antigens, because we will have the exposure during the feeding that is going to happen all the time, even if you have a vaccine. And to enter in our work, we are working with tick salivary proteins obtained by salivary gland transcriptome. So these two papers I'm showing you here in the right side, they are the ones that we use as a reference in our work, the ones that describe the tick salivary salivary gland proteome. And in these works, our group have identified previously around 11,600 sequences in tick salivary glands, in glucocephalus microsalivary glands. And from these, 3,600 are from secreted proteins. So we can see that if we are talking about salivary proteins, we have a lot of options that we could choose to be our target vaccines. And in the past, we did a lot of research and a lot of thinking and talking, and decided on 80 sequences to start working. And these 80 sequences were patented for vaccine tests. And I'm going to show you results from our lab regarding some of these sequences. Okay, so from these 80 sequences, we decided to work with 18 available plasmids that we had in the lab to start with. And from these 18, we were able to produce 9 of the proteins in our lab in a recombinant way. So we had several indicated functions, predicted functions, since we have null proteins that are heavily expressed in all the tick life stages. And we have some null functions like fixation and antimicrobial peptides and evasin proteins. So we selected these, actually these 9 proteins were the ones from the 18 that worked, we could express that. But if you look here in this table, we can see that they cover most, all of the tick life stages, and mainly the male tick, because we do believe that male tick, from our previous results, it is important to support female tick feeding. Okay. And after we produced these 9 proteins, we did it in E. coli, and using a quite standard protocol of recombinant protein production and his-tag purification. Some of these proteins were insoluble, so we use urea. And what we did after is that we were able to actually do the immunization experiment and test the protection of these 9 proteins. So what we did, we used 50 micrograms of each of the antigens. They were adjuvanted with aluminum separately, and we applied the antigens individually in each animal. So you can see here in the red, the dots, the positions. We applied the proteins. They were not put together in a cocktail, so we could avoid some interactions since we didn't know what was going to happen. And we use males for this experiment. We randomized the male before the study, so we got information from previous infestation, counted the ticks, and then put similar animals in each group, so we could avoid some bias. And after the immunization, we did infestation tests, open infestation tests at day 102 here actually, and 162. And between these two infestation tests, we didn't perform a new immunization. So the results we are going to see are about both infestations without a new doses of vaccine here. And actually, our most prominent result was on the number of female ticks that engorged in the end of the experiment. We can see here in gray the cumulative count of ticks in the control group, and here the cumulative count of ticks in the immunized group during the whole of the time we collected the ticks. We collected detached ticks, so we waited them from finish the feeding. They naturally detached and we collected them. And we could observe actually a 62% of reduction on the immunized group compared to the control group. And we could observe a reduction of the detached ticks number in the animals, in the individual animals. So we had some animals in the control group that had more ticks, and some that had less ticks. And actually, this shows us that the animals are quite diverse in the response, since animals in the control group had a few number of ticks also, even if they are not vaccinated. We had some effect on egg oviposition and weight of eggs and weight of larvae. In the end, we calculated the vaccine efficacy of 70%. And after the second infestation, without a new vaccine doses, we had higher reduction in cumulative count of detached ticks to 71%, and the final vaccine efficacy of 78%. Okay, so if we look at the antibody levels, we tested the antibody levels for all animals for each protein, each of the 9 proteins. You can see here that what we observe is that after the third doses of the vaccine, we have significant levels of antibodies increased in the immunization in the immunized group for all of the proteins here in this first point here. And then we have most of the time a slight or a little bit profound decrease before the infestation, and some recovery of the antibody levels in the first infestation. So we can see there are some recovery here after the infestation, after the ticks encounters our immunized animals. Okay, so this happens with almost all the antigens. Just one antigen doesn't present this process. But then when you look at the second infestation here, where we have this tick, and we follow the animals until tick detachment starts, we cannot see for all the proteins actually an increasement again in the recovery in the antibody level. So we do know the animals are protected, but we are not seeing an antibody level increasing with these new infestation. So maybe the point here is not antibody levels, but some information on the quality of the antibodies, maybe affinity, something that we are still looking into. But I like to show this result, to show that maybe the simple antibody level is not really a good measure of protection, since our animals were protected and they were even more protected after the second infestation. Okay, so then we had this multi-antigenic vaccine with multiple proteins that we had 9 shots in one animal. The proteins were separate. This is quite difficult to produce, 9 fermentations, and we do know this is not okay for the industry. So we're trying to move a little bit with this, and we starting actually to work with multi-protein constructions. So to facilitate this vaccine production, we designed 2 multi-protein constructions, and each of them had 10 antigens. So our 9 antigens previously tested, and a 10th antigen that was previously tested by our lab in another moment. And we have these 2 constructions here. Okay, they have the 5 antigens in the same plasmid. So we have 2 fermentations. What it's better for the industrial production. And we have here the model of these proteins. Our idea is to put each antigen with a small spacer between them, a rigid spacer, so we can avoid interactions. And by the modeling, we can see that most of the proteins do not interact, but we have some interaction between antigens 4 and 5 in construction 1 and antigens 2 and 3 in construction 2. Okay. We produced these proteins in E. coli. We have tried in Pichia. We were not able to obtain any result of secreted proteins using other plasmids, so it didn't work for us. For tick proteins, for salivary tick proteins, the Pichia model hasn't worked at all. And we were able to produce these proteins in the E. coli model and to do the purification and on column refolding of these proteins. And this is an easy step for industrial production to do the on-column refolding. And the gradient elution, we were able to obtain these proteins quite clean for immunization tests. Okay, so we already tested these proteins by ELISA with our previous sera from the animals we have tested before. We could see that the animals here, we can see in these two data points, that the animals are able to recognize both construction 2 here, the immunized animals, and the immunized animals can recognize construction 1 also. It looks like a little bit less construction 1, but again, we cannot use just the level of antibodies to define if this will be good or not for vaccination. But we don't know that our vaccine is at least presenting peptides in a way that they are recognized by the animals immunized with the individual antigens. So we have produced these proteins now in the last few months, and we should be starting a vaccination trial in the next weeks. And I hope that if we have another meeting in the following year, we can talk about these results. We have another approach to develop these multi-antigenic vaccines that are the B-cell and T-cell epitope based constructions. So Dr. Mosqueda already told you yesterday about how difficult it is to produce a vaccine for complex parasites as ticks, and that we are probably not going to be able to control the tick with just one antigen. And it's quite difficult to produce big proteins with a lot of antigens. So we also tried the system with identification of B-cell and T-cell epitopes, and just to recover. So the epitopes are the regions of the proteins that are in fact recognized by the antibodies or by the CD4 or CD8 TCRs, right? So we have this from a big protein, we have just a little bit of amino acids that are actually recognized. We have epitopes that are not continuous. We have they recognize specially separated amino acid in a structure, or they are continuous. There are one amino acid is close to each other, and then we have continuous epitopes, peptides for APC presentation by MHC1 or MHC2. And in our group, we work just with MHC2 presentation because we want to increase antibody responses, okay? So because we need both of the responses, both B cell responses and T cell responses, for T cell response, to generate antibodies that are effective against ticks and memory, we have been working on both B cell epitope mapping and T cell epitope mapping. So our strategies for B cell and T cell epitope mapping, I'm going to present them a little bit now. We are applying three main strategies. So the linear B cell epitope mapping by peptide microarray, where we use the sera from the animals previously immunized with the individual antigens in a high density peptide microarray, and we compare the pre and post immunization serum to check which sequences are being recognized by these animals. So we could choose the sequences, use only the sequences to in a new vaccine, in an epitope-based vaccine. Okay, this is one of the strategies for linear B cell epitope mapping. The other strategy we are developing, and I'm not going to talk about this today because we don't have results about it yet. It's the mapping of linear and conformational B cell epitope by next generation phage display. So in this system, we use a randomic phage display library and the serum of our animals, and we collect the phages that are recognized by our animals. And because we use next generation sequencing, we can actually recover hundreds to thousands of sequences to define possible conformational and linear conformational B cell epitopes. And these are a lot of sequences, but after that, we can align them together and try to find the motifs that are actually recognized by the antibodies. And again, I hope that in another meeting we can talk about these results. We are really excited to see what we are going to have from this experiment. And then the other strategy I'm going to talk about to you today, it's the CD4 T cell epitope prediction using data from BOLA GRB3 ligands. So this is a work we have done in collaboration with the Rose Institute and Oxford University and DTU. And I think that this is something, it's a really good tool that you can all use. And I'm going to show you this a little bit later, okay? So to the peptide microarray, high density peptide microarray, what we did is that we designed slides with our proteins divided in 15 amino acid intervals with a 14 amino acid overlapping, so it's the maximum coverage we can achieve in this model. And then we put our serum in contact with this platform, and we check in which spots are being recognized, okay? So you can see here, just to have an idea, how the pre-immunization serum looks like in the slide. It's really dark, we almost don't have recognition. But then after the post-immunization incubation, we can see clear regions here and here and here with several peptides that are recognized side by side, and that are actually our epitopes, our B cell epitopes. And if we generate a heat map of these epitopes, I'm showing you here from one of the antigens, we can see here the heat map for the control groups, for the control group and immunized animals. So each line is one animal. We can see clearly that we have some hotspots of recognition that it's just for the immunized animals. And if we check on these hotspots and we map them against the 3D structure of the protein, we can see that they are located actually in the surface of the protein, right? So we have a 3D model of our antigen 9, and these blue and orange regions are the amino acids that are actually recognized as epitopes, okay? So we did this for all of our proteins, and we were able to identify the main regions of that were actually recognized by the protected animals. It varies a lot between antigens. And then we selected 30 epitopes to do epitope-based construction. And and then we have this result waiting for us to do the final construction. We are going to use the data on the next generation phage display also to do this construction. So we are still going to need a little bit of time to produce this protein and test them in cattle, okay? But we can see clearly that the platform works. It's quite straightforward, and because it's high density, we can test a lot of proteins. And I really support people using it. It shows really good results. And we have the project on the CD40 epitope mapping. So just to put people in the same feed, we do know that to be a T-cell epitope, one peptide needs to be recognized by TCR in T-cell, but to be recognized, it needs to bind the MHC, okay? So instead of looking for TCRs and peptides bind to TCR, we look for MHC class 2, in our case, biology RB3, and the peptides bound to biology RB3. So we developed this project that generated this tool, NetBola2Pen, to analyze biology RB3 ligands. So it's specific for cattle. And the workload, it's quite straightforward. This is not from our paper, but it shows perfectly what we did. So we used bovine cells as models, PBMC, and Tellura infected cells as sources of peptide biology RB3 complexes, MHC complexes, and then we purified these complexes and isolated the peptides. So we had the biology RB3 immunopeptidome, and analyzed the data by mass spec. And then after the motif deconvolution, we were able actually to identify the ligand motifs for several biology RB3 alleles. In fact, we did it for 7 alleles. And we used this information we had the Tellura infected cells, but we used information on bovine peptides. So I'm telling you this because even if you use another infectious model, like Dr. Silvina she has a current project that is quite similar to this one, that works with not the ticks, but the transmitted anaplasma, as I think, or babesia. But the idea is to do the immunopeptidome. You can find information on Tellura or babesia or anaplasma peptides, but you should also use the bovine data to define these motifs. So we can actually use this information if you have new projects running in this area to increase the potential of the tool. Okay, so it is an invitation for people that wants to work in this area to in the future share this data, so we could increase it, make the tool better for everyone. And this is the paper about the tool. I would invite you to check on the paper. And these are the motifs we identified by mass spec and motif deconvolution for 7 alleles. And these 7 alleles, they cover 75% of the biology RB3s, the RB3 alleles expressed in host and cattle. Okay, So it is quite a strong tool for hosting cattle, and we are currently working to make it better with new information, new alleles in the future, and we also count on information of other groups to make this tool better. And here is the tool. It's free to use, so please go there and check it. It's in in the DTU website. It's quite straightforward. You put your sequences here, FASTA or peptide. You select the biology arbitrary from MS method and select all the alleles you want to test, and the result is quite straightforward. It gives you the main ligands. Okay, so we did it. We did this test in our proteins, and we were able to find 7 strong ligands in all of our proteins. It's not much, and because of this, I'm going to show you some result later. Okay, but I would like to invite you to test it too. And after we did all these processes, we selected B cell epitopes and T cell epitopes identified in vitro in silico. And we did a lot of multi-epitope constructions in silico, okay? And we evaluated these multi-epitope constructions all in silico to define linkers and expression characteristics. And we ended up with more than one protein, but we had one that it was the most soluble one, the most okay one to express. It was more stable. And we did the modeling of this protein containing epitopes from several proteins, B cell and T cell epitopes from several proteins. It was nice to see that a lot of T cell, CD4 T cell epitopes were actually overlapped with B cell epitopes. This is why we don't have more than 20 epitopes here. And after the modeling, this is what our protein looks like. So we are going to use the next generation data to design the final vaccine and then produce it. So, and we are now looking for a partner, industrial partner, to go with this project. So if you are interested, or if you know someone that are interested, we are open to discuss about it. Okay. And just to finish this presentation about with the idea of how you can actually use earlier in your project the NetBola Pen tool, I would like to show you some results on CD4 T cell epitope and how to use this data to select antigens. So if you look here in this data from Garcia, this is the cellular transcriptome of the different stages of ticks, right? Fed on hosting. And we can see that we have a lot of family proteins, and inside of the families, we have several dozens to hundreds of proteins, okay, in the same family. They have the same or similar motifs that could be blocked by antibodies, but we ask ourselves if due to the protein redundancy and this great number of proteins, how do we choose between them? Even if they have the same functional motif, and we want the antibody against that functional motif, it is enough to select one protein. This one protein, it is enough to induce CD4 T cell responses in the long term and for all the animals, and memory responses. So what we did is that we did this test, this in silico test, with some of the most expressed families in our cellular transcriptome. We used lipocaline, protease, and cement families. You can see here that lipocaline family had 294 members, so it is impossible to just look at this family and select some antigens. We need to check on them carefully. And just for the exercise, we selected 10 of the most expressed proteins in each family and analyzed them with NetBola 2 pen against that 10 alleles to check how many proteins we needed to be able to have 100% coverage of all alleles. So we could immunize the animals in a herd and all the animals would have CD4 cell stimulation for memory and for antibody production. And we could see that actually for the lipocholine family, we had some proteins that were able to cover to have ligands for 6 of 7 alleles. So this is quite a lot. It is good. But for some other proteins, as protease family and fixation proteins, we actually don't have a lot of coverage. And in fact, some of the proteins have just were just recognized by one allele. So if we choose one of these proteins for vaccines, we may not be able to induce antibody response to all the animals. And again, if we consider that the animals are quite somehow inbreed, this is not the right word, but we have a population number that is quite low. If we lose 2 or 3 alleles, it is already quite a lot. So if we want to have the maximum coverage in these proteins, we would need actually to use 2 lipocalins in our proteins, 2 cement protein in our products. And for the protease family, there was no possible combination that we could achieve cover from all of the alleles. So I think this is a really good and nice way of looking at your proteins before developing your vaccine project, to select the antigens using a cattle dedicated tool, the NetBall 2 pen, and to help you to decide which proteins you should use. You could use from same families. Okay. So this is what I had to show you today. I would like to thank my partners in the Rhipicephalus microplus team here in Dr. Beatriz Ferreira lab, especially Alexandre de Moraes and Mauricio Peña, that were the people that generated data on the epitope-based vaccine and multi-protein vaccine. And I would like to thank also the team that worked with the immunopeptidomics and immunoinformatics to develop NetBall pen. So Dr. Tim Connolly from Edinburgh and Nicola Turnett from Oxford and Morten Nielsen from DTU and his student Birger that did all of the work in the immunoinformatics. Thank you very much, and please keep my contact if you want to talk about something or need help or can help us. We are open to discussion. Thank you. Yes, I'm not hearing you. Sorry.
Yeah, I'm sorry.
Okay.
Thank you very much for your presentation. There are several questions in the commentary section. One is, there are some by Dr. Carlos Vega, and he's asking, it is understandable that peptides might be antigenic, but will they be immunogenic? That is, how would they protect cattle from ticks?
Yes. So, yes, this is our next question to be answered, actually, because we haven't tested these constructions in cattle yet. We are going to test immunogenicity in mice at first, just to check if they are really inducing responses. But we have a test. We have to test them in cattle. It is possible that some of the peptides of the epitopes are not immunogenic, and we have to find a way to check these if they are protective or not. And if they are not, maybe we need to change the platform. So I've shown you the recombinant protein structure, but we are working with the idea to put them on VLP or liposomes or dendrimers. We are still discussing this to make them more immunogenic. So I hope they are, but it is a limitation.
Mm-hmm. Probably because of one of the platforms that you showed where some of the peptides that you analyze were identified by antibodies. But did you go back and see whether these are real peptides in the tick?
They come from transcriptomic data, and these same proteins were previously proven to be presented in tick saliva. They were present in tick saliva by MS. So this was one of our first steps, really in the beginning before protein before we patent the proteins. So they should be, but it would be interesting to get specific antibodies and check on the tick if they are really being presented now. Yeah.
Another question is once you establish that these are immunogenic, how often do you need to be? How often do they need to be applied?
We will need to test that, because if our idea that the saliva per se it is a natural boost, maybe we don't need to vaccinate that much. We did 3 doses in our test, and after 4 months we had the same protection. We need to go for this test for longer to check if it works. So we don't have any idea on how this is going to work. We will need to test that.
Jose Luis Ortega says, congratulations, Dr. Andresa, excellent conference. I agree. And Dr. Carlos Vega says, do they work the same in both Taurus cattle as well as in both Indicus?
We haven't tested that. We just used hosting animals by now. We should be working with a mixture now of hosting and gear, but a part of that we haven't tested in other animals. It would be really interesting, because we had a previous talk with another researcher here that tested a vaccine for babesia or anaplasma. I don't remember now, but she tested in 2 different breeds, and the responses were different. One breed was protected, and the other one wasn't. We don't know why this happened, and maybe there's something to do with the CD4 T cell epitopes. So this is a big thing, and we are currently working on funding to elucidate some of the features of the CD4 responses and ligands in different breeds of cattle.
Yeah, another question here is probably, is there some work going on vaccine for dogs? I assume it is for ticks.
Yes, there is. We actually did a test previously with microplus antigens in mice infested with sanguineous. It worked in mice for individual antigens, but then in the past we tested the same 9 proteins in dogs, and we had some protection, but not really good protection. We had just a little bit less engorged tick females. So we need to probably to work on specific data from sanguineous transcriptome to get a good sanguineous vaccine.
Okay. Dr. Joseph Bush is asking, is there an optimal number of antigens that can be used in a single vaccination? Is there a maximum number?
I would say no. Actually, it depends a lot on your platform. So in E. coli, we were able to express these proteins up to 100 kDa. So if you could put like 10 proteins in this size, small proteins, or maybe 2 or 3 that are bigger, that's okay. It depends a lot on your platform. We had this. We were afraid in the beginning that maybe they would compete a lot, but it looks like it's not case. And Dr. Mosqueda showed us results yesterday about epitope-based vaccine with a lot of different epitopes, a chimera, and the vaccine was protective. So I think we have increasing evidence that the number of epitopes is not a biological problem. It can be a platform technological problem, but not a biological problem.
Mm-hmm. Okay. I have a personal question for you. Is it difficult to test the biological function of each single peptide before ensembling your multi-antigenic protein?
We could, yes, put effort on that. This is one of the things we try to do at the lab. But the model is really difficult to work with the tick saliva, and we would have to generate specific antibodies for each of the peptide, and then use it in really specific models and collect saliva to test it. This is quite difficult because we don't have this structure here. I do believe that other groups that already work with some functions could actually test this better, like groups that already work with metalloprotease from that have a system already established could actually test these functions and check if it works. So it is difficult in our reality, but it's feasible. It's something like Dr. Mosqueda said yesterday about establishing rhipicephalus sanguineus work in the lab. It is quite difficult because you have to keep the ticks and there's a lot of work involved. So I think there's a lot of work involved, more than biological issues.
Mm-hmm. Well, I think these are all the questions that we have, which I think
Thank you.
Great. Thank you very much for your presentation, and I'll see you soon.
See you soon.
Bye.
Bye.
Now I want to take this time. I don't know if we have time for a video of our of the Elanco. ¿Podemos pasar el video de Elanco ahora?
Elanco acaba de expandir sus horizontes. Ahora, toda la línea de productos de salud animal de Bayer, reconocida mundialmente por su alta tecnología y calidad, hace parte de nuestro portafolio y llevará a nuestra marca por nuevos caminos. Es así, expandiendo su participación y seguimiento, que Elanco consolida su liderazgo e innovación en la prevención y tratamiento de animales de producción y animales domésticos. Bienvenido al movimiento Elanco de crecimiento saludable de las relaciones de negocios, pero lo más importante, de la salud y el bienestar animal. Elanco, porque crecer es saludable.
Bueno, pues nuevamente agradecemos a todos su presencia y les recordamos que esta transmisión está también siendo realizada en el canal de Facebook en español con traducción simultánea y en YouTube para aquellos que quieran escucharla en inglés. Now we are going to present our next speaker, and he is Dr. Masaru Ueti. Is he around? ¿Está por ahí el doctor Masaru ya? Show. Y estoy confundido, no sé si toca el tiempo de Masaru o de Joe.
Juan, can hear me?
Yes.
Can hear me?
Masaru, yes, I can hear you.
Okay, I was in wrong room.
All right. Okay, everything is set now. So now
So
I'm
let
gonna.
me share my video to see if it's working. One
All
before
right.
you go, okay?
Yes.
Which monitor? Share. Can you hear me? Can you see?
Yes.
Okay, very good.
Excellent.
Sorry for the confusion.
No, don't worry. It is all right. So I'm gonna present you Masaru.
Mm-hmm.
Dr. Masaru Eti received his DBM in 1994. He's also a veterinarian, and his master's degree in 1997 from Londrina State University, the School of Veterinary Medicine in Brazil. Dr. Ueti completed his PhD in 2005 at the Department of Veterinary Microbiology and Pathology at Washington State University. Since 2009, Dr. Masaru Eti is a research veterinary medical officer assigned to the United States Department of Agriculture Animal Research Service Animal Disease Research Unit in Pullman, Washington. His main research topics are the development of vaccines and diagnostic methods for tick-transmitted diseases, vector biology, and pathogen tick mammalian host interactions. Thank you very much, Masaru. Go ahead.
So was very nice before my presentation. See, presentation from Brazil. I'm very glad that, and was very nice presentation. I'm glad to hear that you are doing a very good job in there with ticks. Today I'm gonna discuss with you a little bit different from the ticks, but it's important that the ticks transmit the parasite. I'm gonna discuss with you Babesia stage-specific proteins as a candidate for prevent disease or broken parasite transmission. For those that don't know me, my name is Masaru Eti. I work for the United States Department of Agriculture, Animal Disease Research Unit in Pullman, Washington State. The mission of the unit is to solve basic and applied problems concern persistent infectious diseases of livestock. My laboratory focused on the development of strategies to control tick-borne pathogens, specifically hemoprotozoa parasites of cattle that cause bovine babesiosis. I'm going to take a little time here to tell you one statement that I read in 2013. That is, in the current advance in understand the biology of Babesia bovis. In that article, they mentioned that overall, advances in our understanding of the biology of the parasite, in this case Babesia, itself has been slow and arduous, with only a handful of research groups worldwide taking up the challenge to shed light of this hemoprotozoa parasite. This is true in 2013, and it is true today. And I hope that with this presentation, we'll be able to stimulate collaboration between different groups to improve our understanding in parasite biology and development of control strategy for bovine parasites. So why we study bovine babesiosis? That is the big question. So bovine babesiosis is one of the most prevalent tick-borne diseases of the livestock. It is worldwide distributed in tropical and subtropical regions. Babesia bovis and Babesia bigemina are distributing Asia, Africa, America, Europe, and Australia. And they are correlated with the presence of rhipcephalus ticks. In contrast, Babesia diversion is restricted to Europe, that where the exotic ricin, a competent vector, is present. Bovine babesiosis caused significant economic losses for the livestock industry. In Latin America, more than $800 million were lost last year caused by bovine babesiosis. And these losses are associated with high morbidity. Young animals often survive, adult animals often die. High mortality. In case of Babesia bovis, it caused cerebral babesiosis. Abortion, reduction in milk and meat production. What are the challenges to improve animal health concern bovine babesiosis? One is the limited knowledge of mammalia host vector and parasite interface. Limited available intervention strategies. There are few strategies to control the disease. In this case here, example is live attenuate vaccine. However, not many countries use the live attenuate vaccine, and there is no subunity vaccine to control infection or transmission of the parasites. Acaricide, tick population start to have resistance to acaricide, also for multiple acaricide resistant. Ticks are very efficient to acquire the parasite from acute, that means parasitemia above 10 to the 7 red blood cells, infected red blood cell per ml of blood, or low parasitemia, below 10 to the 6 parasites per ml of blood. The parasite transform within the ticks, develop in the hemolymph, transfer to the eggs, and the offspring of these ticks are infected, and when they feed on naive animal, they transmit, and the parasite infect the red blood cells and cause acute diseases. When infected ticks feed on naive animal, they inoculate the sporozoites, and the sporozoites infect the red blood cells. In case of young animals, the animals survive and they become persistently infected for life. When the ticks feed on naive animal or infect animal, they ingest the parasites, and the parasite transform in sexual states in the lumen of tick medagut. The gametes, macro and microgametes, fuse to form the zygote. Zygote infected the medagut epithelial cells, transform in kinetes, and kinetes are released to the hemolymph. And these parasites infected ovarian epithelial cells, transfer to the eggs, and the next generation of ticks are infected with babesia, and when they feed to naive animal, they inoculate the parasite, and the parasite complete its life cycle. Today, I'm gonna focus on in few areas of the development of babesia. The parasites that develop in the blood states, in the blood of the mammalian host, the parasites that develop in the lumen of the ticks, and the parasites that develop in the hemolymph of the ticks. We are discussing this parasite state in the context of gene and protein expression to develop a strategy to prevent disease or to develop broken transmission vaccine. So to do this type of experiment, we develop the in vitro culture of Babesia bovis blood states, here indicating these figures below. We inoculate the animals, splenecto animals, and collected infected blood with a hyperesthemia. We isolate this parasite for proteomic and transcriptomic. We use these hyperesthemic red blood cells, and we induce the parasite in vitro to develop in sexual states. Here, show the development of sexual state, macro and macro gametes. Then we also develop a system to isolate kinites from the female ticks using the ex vivo Babesia kinites. We put the ticks to feed in high parasitemic animals, collected the engorged female, and then extract the kinites from the hemolymph and use these kinites to test our hypothesis. So the approach that we use in these experiments are the proteomic, the 2D differential in-gel electrophoresis. We use Babesia bovis blood state and kinete state. For the transcriptomic, we use RNA-seq to identify upregulation, gene upregulation. We compare Babesia bovis blood state and kinete states, and also we compare Babesia bovis blood state and sexual states. And then we validate our finding proteomic and transcriptomic using protein expression by live and fixed IFA. We use blood, Babesia bovis blood state, sexual state, and kinates to determine whether or not the proteins are expressed by these states. So for the proteome, we inoculate an animal with stabulates, put the ticks to acquire the parasite, and then we isolate Babesia bovis blood state and kinete state. We mix or we label with different wave dyes, each states, interrupt the reaction, and mix all the protein together. Running 2D gel, find the spots that you are interested in, send to mass spec, and determine which protein, which genes or protein is expressed by kinete parasites. Here is the result for the proteomic. Let me go through this and orient you for this result. So A is the comparison between Babesia bovis kinates and Babesia bovis blood state. B is the comparison of Babesia bovis kinates and tick hemocytes. And C, Babesia bovis blood states and tick hemocytes. So in A, the red indicate Babesia bovis kinates and green Babesia bovis blood state. In B, again, Babesia bovis kinates in red and hemocytes in green. And C, Babesia bovis blood state now is the red one, and tick hemocytes is the green. Yellow indicate common proteins between the states and the ticks hemocytes. So what we found here that when we compare Babesia bovis kinetics and blood state, we found a spot that suggests that is kinase specific protein. We look at change the wavelengths and compare Babesia bovis kinetics and tick hemocytes, and the spot is still in the same place. When you compare Babesia bovis blood state and tick hemocytes, there is no evidence of the protein expressed by Babesia bovis and tick hemocytes. Suggests that this spot here are Babesia bovis kinase stage protein. We collected these spots and submit to mass spec, compare to the genome, and identify several spots that contain different proteins. So here are several proteins that or genes identified, and then we are very interested in this BBOV underscore I002220. And from now on, I'm going to call this as a 2220. And why we are interested to work with this protein is have a signal peptide, indicate that potential is a surface molecule, is transmembrane, have a transmembrane domain, a small protein that is easy to express, and the most important, this protein here have only homolog with Babesia bigemina that we call true Babesias. There is no homologous to Babesia microti, Teleria annulata, Teleria equi, Teleria parva, or Plasmodium parasites. There are other protein here that, another protein that we are interested in, this BBOV 3400. Have a signal peptides, transmembrane domain, and have a homolog to Babesia bigemina, but also have homolog to Teleria parva and Plasmodium parasites. So if you want to, I'm going to focus more in this 2220. If you want to have more details about this, the others protein, please look at in the Journal of Proteomic Research that we published in 2017, discussing all these proteins. So then we want to verify and confirm and verify whether or not what we found in the proteome is correct. And the proteome gave a very limited numbers of protein to work in our system. We want to determine using RNA-seq to see if you find additional proteins, genes or proteins that can be added to our research. And for the RNA-seq or transcriptome, we infected the animal with Babesia bovis. We put the ticks to feed on to engorgement and collected kinases. We collected blood from infected animal, growing culture, and isolate the Babesia bovis blood states. We extract RNA and submit for RNA-seq to determine differential gene expression. In our high throughput RNA-seq, we compare Babesia bovis blood state versus kinase state. And if I'm going to talk to you about all the data that we collect from RNA-seq, we will stay here for maybe 2 or 3 days. So I'm going to just focus in very few questions and that we have in this for this presentation. And in the RNA-seq, we found that 28% of the proteins are upregulated in the blood, 26% of the genes were upregulated in the kinates, and 46% of the genes have no significant difference between blood state and kinase state. We found that 1,096 genes were upregulated in the blood state with fold chains from 2 to 300 difference between kinates and blood state. We also found that 997,000 genes were upregulated by the kinates. The fold change vary between 2 to over 1,000 fold differences. We want to confirm whether or not our RNA-seq is giving the correct information for us. We look at, in the RNA-seq, MSA gene family to determine whether or not they are upregulated in blood and kinates. We know that MSA is a gene and proteins that are important for the parasite to invade, replicate, and survive in the blood of the mammalia host. When you look at these genes, the MSAV gene family, we found that all the genes, MSA1, MS2A1, 2A2, 2B, and 2C, are upregulated in the blood state, but not in the kinates. In the kinates have very low or no existent presence of the transcripts. We have another gene here that we want to compare. The 1680 show that it is upregulated in the blood state, but low in kinates. We also look at the genes that are expressed during tick infection. We know that the 6Cs gene family is important for the development of Babesia within the tick midgut. We have been published that the 6Cs-A and 6Cs-B is high expression by the gametes. We compare the 6Cs gene family gene expression in kinates versus in blood state. As you can see here, all the 6Cs family are upregulated in the kinates and low expression in blood. Again, we look at 2220. Remember that in the proteomic, we found that protein expressed only by the kinates. And here we see that the 2220 is upregulated in kinates and down in blood state. Corroborate with our previous study with proteomic. So this is some examples of the genes that are upregulated in the Babesia bovine blood state. Some of the genes here, such as SMORPH, are very highly transcribed. And you have VISA that are important for the cytoadhesion of the parasites using red blood cells. Another is SMORPH. another visa, we found a putative integrate proteins. Again, several other SMORPH, MSA1, MSA2A1, spheral body proteins, putative integral membrane proteins, another visa. So very consistent what we expected that Babesia above blood state express. The negative log here means that they are upregulated in the blood state. And all these genes have a significant difference between blood states versus kinase state. So let's look at some example of Babesia bovis kinase gene regulation. So we have here 2220 that we call kinase specific protein, KSP, and is the highest expression transcribed at very high level. Then you have a TROP1 that is also very high transcription level. And interestingly is the TROP1 in Babesia bovis have been postulate that is important for the motility of the merozoites. When we compare this protein, TROP1, with kinase and blood state in hours isolate, this protein is only expressed by the kinase, but not by the blood state, also is not by the sexual state. We have 6Cs, C, that have a very high transcription level and important probably for the development of the parasite within the ticks. Another 6Cs here, A and B, and have been postulated that these genes are important for the sexual development and fusion to form the zygote. We have AP2 domain family, very high expression. And again, all that have the significant difference between kinase versus Babesia bovis blood state. So the next experiment that we did the high throughput RNA-seq comparing Babesia bovis blood states versus sexual states. And what we did in this experiment, we inoculate splenectomized calf to have a hyperostemia, collect the blood from this hyperostemic animal, culture to develop blood state, and then we induce sexual state using xanthurinic acid. Here is the example of sexual state development. We isolate RNA from blood state and RNA from sexual state and submit to RNA-seq. And what we found was 854 genes were upregulated in the gametes, 790 were upregulated in the blood state. These 790 upregulated genes also match with the kinase versus blood state, that they are upregulating the blood versus the kinase, and the same genes are upregulating the blood versus the gametes. So this is an example of our find. We just finished this 2 weeks ago, and we need to analyze the rest of the data. But a preliminary data showed that the 6 cis genes are upregulated in the sexual state. That's what we expected. Here are the fold chains, vary between log 2, 0.4, 3.2, 3.9, 2.9, and go on. And we have the 2220 again, KSP, is also upregulated in the gametes. GCC2 and GCC3 that we found that they were upregulated in kinase stage also is upregulated in gametes. And HAP2, that is important to the fusion of the gametes, is expected to be upregulated by the gametes and low in the blood state. Indeed, the HAP2 is upregulated in the gametes and low in the blood state. So to verify if this protein and the location of the protein expression on the parasite, we use live IFA to determine if they are surface exposed protein or fixed IFA using kinase. We use fixed IFA because it's very difficult to isolate kinase, live kinase. We generate antibodies against peptides and use these antibodies to determine the location and the expression of the protein by the states of Babesia bovis. So here we show that in the sexual state, HAP2, 6Cs, KSP, GCC2, and GCC3 are expressed by the blood state. Here show the, sexual state. Here show the DNA labeling. Here's the antibody reactivity and the merged image between the nuclear labeling and the antibody labeling. And these results show that these proteins are expressed by the sexual state on the surface of the parasites. Then we look at if the protein also are expressed by kinases, specifically for KSP and GCC2 and GCC3. And then what we found is we label with DAP, the nucleus, the antibody, and the merged imaging. And we found that this protein is expressed by the kinases, but not the blood state here. We look at GCC2 and GCC3, and very weak reactivity here, as you can see, but not reacting with blood states. We don't know if these proteins are expressed on the surface of the kinase, due to the difficulty to isolate live kinases for this experiment. In conclusion, we were able to determine gene regulation by Babesia bovis parasites, blood states, gamete states, and kinases. We demonstrate differential protein expression by Babesia parasite, specifically for gametes and kinases that can be used for vaccine development. What is the impact of this research? Is the development of vaccine to prevent the disease, in the case here, using antigens to the blood state, and or transmission block vaccine using proteins from gametes and kinases. At this point, we are in the position to start to test our hypothesis, looking for proteins and generate proteins as antigen to determine whether or not those proteins, such as MSA, SMORPH, spherobarb protein, can protect the mammalian host against the infection of the parasites. Also, we are in the position to test the hypothesis whether or not immunize animals with protein expressed only in the sexual state, such as HAP2, 6Cs, and other protein exclusively expressed by the sexual state, can be used to immunize the animals. An antibody against this protein, when the ticks feed on the infected and immunized animal, can block the interaction of the sexual state and prevent the formation of zygote and infection of the mid-gut epithelial cells. Also, we are in the position to start to develop proteins, recombinant proteins, to immunize calf and test whether or not specifically 2220 can be used to immunize these animals and test whether or not they can block the infection ovary and consequently prevent the transmission of Babesia bovis to larvae, and then prevent the transmission of Babesia bovis to naive animals. Alternative to all these specific area, we could use all three areas in the blood state, in the sexual state, in the kinases, proteins to immunize the animals and prevent disease manifestation and transmission of Babesia bovis to naive animals. I would like to thank you, several people in my team here, Dr. Carlos Suarez, that have been doing a great job with testing some hypotheses, specifically HAP2, and there is a few experiments that he need to finalize, and there is a promising result with HAP2. Dr. Naomi Taus is the one that is working with immunology and help us to develop the strategy to determine whether or not the proteins are important for prevention of infection of the mammalia host. And Wendell Johnson, Michelle Musso, Lowell Kapmeyer, David Harden, Paul Lacey, and my postdoc, Hala Hussein, and Janaína Capelli Peixoto. And this research was supported by USDA Intramural Grant and USDA NIFA Grant. Thank you very much for your attention. I'm glad to take a few questions.
Thank you, Dr. Massaro, for your presentation. I don't see any questions yet, but I would like to ask you whether I've seen from your results that you've characterizing many antigens from kinates and sexual stages.
Mm-hmm.
And what is your idea for those? Are you planning to choose one or two, or are you planning to use them as a multi-antigenic vaccine, or what would be the idea?
So let's start with the sexual state and kinates. Okay. So sexual state, what we plan to do is to test individual antigens. The reason is we want to make sure that we found which antigen have efficient control of the development of sexual state. Example, HAP2. HAP2 have been postulated that is important for the fusion of the gametes. And with this, the HAP2, we are testing whether or not they can prevent the fusion, consequently prevent the formation of the zygote of the parasite, and prevent the sexual state. There is one possibility. And then we can start to test other antigen, for instance, 6Cs-A and 6Cs-B, separated, because they are potentially important for the development of Babesia bovis parasite in the lumen of the midgut. And that is the idea. With time, and then the next step after we identify which proteins are important for prevention or reduction of the infection, perhaps we combine all the proteins in the sexual states and try to determine whether or not we enhance the control strategy of antibodies that prevent the infection of the ticks. Alternative also is to look at the 2220 that I mentioned that is very high expressed in the kinases and used to immunize the animals and determine whether or not that protein is important for the attachment and invasion of the ovary. So if you find that have some impact in the infection of the ticks, and if they don't reduce completely, because I believe that there is no a silver bullet, we need to combine different antigens to determine or prevent the infection of the ticks and perhaps reduce the transmission efficiency of Rhipicephalus microplastics.
All right. Thank you. Ivan Corona is asking you, have you tried comparing the specific upregulated genes in each stages which known pathways in all therapy complex of parasites?
So the answer for your question, no. We are in the process now to finalize the sexual states RNA-seq to see which one are regulated and downregulated, and then we are combined to evaluate the way that you are asking to see with others AP complex parasite, specifically for malaria that have much more data. And then we were plan to evaluate this gene's function with the others AP complex. However, in this experiment that we did specifically for kinases, as I mentioned before, the 2220 and there is different genes in there also that is specifically for the transovarial parasites. There is no homologs in Plasmodium that is not transovarial. Babesia microti, Thaleria annulatus, and Thaleria parva. That make a little bit difficult to compare this upper regulation in the overall complex of the upregulation. But I agree with you, it's like a good idea to look at and compare, at least at this point, to see if there are some function related with upregulation. Thank you.
All right. Thank you. Masaru, we are having a lot of veterinary students here attending the presentation. I don't know if you would like to comment on, and also professors of some universities in Mexico. I don't know if you would like to comment on how could they approach you and your group to do some kind of collaboration of research.
So, as I mentioned to you before, there is a very handful of research group who work with this, and I work with close with Juan Mosquera, Dr. Juan Mosquera, and several people from Mexico and INIFAP. Very little numbers of people that have been doing research in bovine babesiosis. My group here at USD, we're open to do research collaboration with any university from Mexico. And important for us is, in the United States, there is no bovine babesiosis. But as a research, we need to keep bovine babesiosis outside the US. The reason is this: if we let this parasite to come to the United States, we will have a catastrophic outbreak in the United States. So, working together with Mexico, we have two things going on. One is prevent the introduction of Babesia to the United States, and also help our neighbor country here, Mexico, to develop a strategy to control bovine babesiosis. And students is welcome to come to my lab to work with me, and professors are welcome to interact with us to develop a strategy to control bovine babesiosis. There are a lot of things that we found in this research here that need to be tested, and only one or two groups that work in this will take about 100 years, and I don't have time to do that. Maybe we work together, perhaps you will speed up a little bit more development of bovine babesiosis. And I always like the idea to collaborate with other institutions that can help us to progress with development of vaccine to prevent bovine babesiosis transmission or disease manifestation.
Thank you, Massaro.
You're welcome.
I have a couple of questions here for you. Dr. Andresa Fish says, great presentation. Do you have several possible antigens? Any system in mind to improve antigen-specific antibody generation to test neutralization in vitro, maybe recombinant antibodies?
Very good question. So the few things that we need to understand first is the system and how we do neutralization. Yes, is a multiple question in there. Let's start from the antigen expression. We are going to express these antigens via eukaryotic system. Why we want to do eukaryotic system? Because we're mimicking close to the Babesia. Babesia is a eukaryotic parasite, and we want to use the same system to express this protein. We are looking first for the bioinformatics to determine which region of that protein can be predicted as a B cells epitope. And then we are using different adjuvants to help us to increase the antibody titers. Second question is that a neutralization. So neutralization can be used when you have a parasite that's replicated in the in vitro system. For instance, Babesia bovis blood stage. We can use some of these antibodies to look at if we are able to neutralize the infection or inhibit the infection. The other system that you have is the in vitro induced sexual state. At this point, we don't know if you can do the neutralization to prevent the formation of the zygote. The system tells us or give results that they can be induced to gametes, but we don't have evidence that this gamete fuse to form the zygote. It would be very difficult to do the neutralization because we don't know what is the readouts for our research. And for kinates, we don't have in vitro system to do the neutralization assay. Everything that we do with kinates need to do ex vivo. We need to infect the ticks. We need to collect the kinates and test all the proteins or genes and proteins from the ex vivo parasites.
Mm-hmm. Thank you. We are running out of time, but I want to ask you another quick question. It says, Diego Josimar says, is there a way to guarantee that Babesia cells that come from culture and were induced to gametes to proteomic analysis are expressing the genes in comparison with in vivo ones?
Diego, thank you very much. Very good question. And yes, you're right. Maybe the induction of the in vitro will not translate what we see in vivo. The postdoc that I have here and that is working with us, and she is looking to generate antibodies, and then what you want to do is to infect ticks, collect the mid gut of the ticks, infected with Babesia bovis in different time points and look at if that protein is expressed by in vivo sexual states. That will give a little bit more information about which genes that we can use for which genes and which protein that can be used for vaccine development. We know for at this point the HAP2 that is expressed by in vitro sexual state and also is expressed by in vivo sexual states. That we know. That is the experiment that Dr. Carlos Suarez is doing to determine whether or not that HAP2 is HAP2, HAP2 is important for fusion of Babesia bovis gametes and prevent the transmission. He has a very, very good data at this point, but it's a preliminary data, and he plans to do another test and to increase the numbers of animals that he used and perhaps have a better idea if this HAP2 is important for vaccine development.
Okay, thank you very much, Masaru. I think we are just on time. I really appreciate your collaborating with our symposium, and we will keep in touch. Have a good day.
You too. Thank you very much.
Bye-bye.
Bye.
Our next speaker is Dr. Joseph Bush from Northern Arizona University. And he is actually working as a senior research scientist at the Pathogen and Microbiome Institute in Northern Arizona University. He's one of his research focus is acaricide resistance in ticks, which is the one that is going to talk us today. Thank you, Dr. Bush. Go ahead.
Great. Thank you, Juan. And is the sound okay?
Yes.
Great. And you can see the first slide.
We can see it now.
Okay, great. Thank you. Hello, everyone. I am Joe Bush, and today I want to present results from our population genetic data on cattle fever ticks, including acaricide resistance movement in Mexico and Texas, but also just looking at how ticks move and some of their cryptic movements on cattle and wildlife. First, I just want to thank Dr. Mosqueda and the organizing committee for inviting me to present today. I am very honored to be here, and I am enjoying all of the presentations. It's fun to learn about all these different aspects of ticks and the diseases that they transmit. So I am a staff scientist at the Pathogen and Microbiome Institute, shown here. My faculty supervisor is Dave Wagner, who is a member of the faculty at Northern Arizona University in Flagstaff. We are up in the mountains in Arizona, up at 2,200 meters. And the mountains in the background that you see are the San Francisco Peaks. So that is where I am presenting from today. So as you can see from the list of collaborators, our projects come out of a collaboration of many scientists at different laboratories, many in the United States, from the United States Department of Agriculture, but also we collaborate with Dr. Mosqueda in Mexico and Dr. Guy Klafke, who is in Brazil. We have been fortunate to maintain funding from the United States Department of Agriculture to keep this work going for the past 10 years. And what are we working on? So the problem are invasive cattle fever ticks. The photo shows Rhipicephalus microplus. As most of you know already, this is a highly invasive tick. It has spread across the globe to many cattle-producing regions of the world in the tropics and subtropics. It is considered to be the most economically important ectoparasite of cattle worldwide because it transmits the Babesia parasites that cause cattle fever. And so this disease is an important problem for cattle producers in many countries of the world, including Mexico. And in the next slide, I show a few more details about Babesia. I don't need to go into many details because that has been covered already very well by Massaro and others. So just briefly, in the Americas, bovine babesiosis is caused by two species, Babesia bovis and bigemina. And as Massaro just explained, it is often lethal in naive adult cattle. And that is why the US is trying to avoid its reintroduction, because our cattle herd has not been exposed to Babesia as calves. And if the US cattle got this parasite, we would likely see a very acute outbreaks. Interestingly, calves usually survive an infection from Babesia and become lifelong carriers. And so ticks, especially cattle fever ticks, can acquire the parasite from a low-level cow and then start disease transmission. This disease and tick vector system have an interesting history. It is the first published and demonstrated example of a vector-borne disease where the parasite was linked to the ticks and was, it was established that you needed the tick to move the parasite. So since there is not yet a vaccine for Babesia in the US, the disease prevention is very focused on controlling the ticks. And so that is what a lot of my talk is focused on, will be on the tick side of this equation. Next slide is to show a little history about tick eradication in the United States. The US is currently free of babesiosis, but it has not always been that way. And this map shows the extent of cattle fever, ticks, and Babesia in the red areas. This is as of 1906, when the eradication program in the United States was launched. It was a very successful program, and cattle fever ticks were eventually eliminated from the southern United States. So an interesting side note is that the early eradication was achieved using arsenic, plus some other ingredients like soap and petroleum tar. And so it was very toxic. Interestingly, it was very toxic, and we would never use that combination of chemicals today. Now, ticks did become resistant to arsenic, this very toxic combination. And so I think this is a demonstration that ticks can become resistant to almost any chemical that is used too frequently. Here is the state of tick populations today in Texas. So the eradication was complete by 1943, but because we continued to see tick populations crossing into southern Texas, a quarantine zone was established in 1960. That's the purple line that you see here, and the purple areas. Wherever you see a red property, those are places where cattle are infested with ticks. So along the Gulf of Mexico, this coast here, you see a lot of infested properties. And then sometimes you will see an infested property far to the north. Around these red properties are surveillance areas. They are buffer zones to try to keep the infestations from spreading too far. So when you look at this, kind of the broad picture, you see this is very much a biological invasion zone. There is a nearby source population in northern Mexico, And then ticks also are often established for several years at a time down in the quarantine zone along the Rio Grande. There is a lot of good habitat. So it is difficult to eradicate ticks completely in this area. But as with any biological invasion, most of the colonizing happens very close to the source. And then there are a few more dispersal events as you get farther north, and only rare events that will go, over 100 miles. So short distance dispersal is more common. So to prevent the movement of ticks, there is a lot of monitoring, especially on horseback, the tick riders. These are APHIS inspectors that are on horseback. They're looking for stray cattle that will come across the Rio Grande. And then those cattle could also have babesia. So that is a very important host to try to remove from the system. What else do I want to say? Just that the goal in the United States is to eradicate ticks. We are not trying to control. We are trying to eradicate all the ticks because of the issue that babesia could have on cattle that have never been exposed. And so I want to show you a few of the challenges for eradication. And one of those is what I just mentioned, that there are cattle on the border that do cross, and shown on the map here are locations where these are the triangles showing where stray cattle from Mexico have crossed and are intercepted in Texas. And then not all of those cattle have ticks, but the ones that do have ticks, we have been able to genotype them and look at some of the genetic patterns. These cattle represent the risk of babesia transmission. And in 2018, Glenn Scholes performed a study where he looked at the blood of these cattle that did come across. And you can see that 1 in 5 carried babesia, either bovis or bigemina or both. 3 out of 5 carried Anaplasma marginale. So he has published those results recently. So that is important to take control of this risk. And then, of course, these cattle can have cattle fever ticks, which there's a risk of establishing populations along the Rio Grande. And then the other important challenge is that the cattle fever ticks can also use alternative hosts, such as white-tailed deer and other species like introduced nilgai, antelope, and red deer. We think of cattle fever ticks as feeding on cattle, but that doesn't seem to be the case whenever they invade into a new region. And hosts like these are even better at crossing the Rio Grande, and deer with ticks and nilgai, they move back and forth across the Rio Grande. So there's ticks moving in both directions across the river. Once ticks are in the quarantine zone, it is likely that these wildlife species are responsible for moving ticks short distances. They're very good at crossing fences. And so they're better at jumping fences than cattle are. And so you get a progression of tick infestations until you get outside of the quarantine zone. And once the ticks are outside of the quarantine zone, they are in the free area and they are not monitored very closely. And then we get long distance movements on cattle. The ticks will get back on to cattle from the wildlife. The final challenge is, of course, acaricide resistance. And so the number of acaricide resistant infestations has increased in Mexico and also in Texas. And the USDA does monitor constantly for infestations of ticks that are resistant. And here's the list of chemicals. A great example of this is resistance to synthetic pyrethroid chemicals like permethrin. They have spread widely in Mexico and reached Texas. And so what are we doing with population genetics? You know, I just outlined the problem. What can be done? Our vision for making a difference with this problem is that genetic tools can be used to improve tick management in southern Texas and other places too where ticks occur. With population genetic data, we can see how ticks move. We can find where they come from. We can see if there's a single invasion source that makes it into the quarantine zone of Texas, or are there multiple genetic groups that move and use the landscape at the same time. So the first data I'm going to show you are from DNA fingerprinting markers. We have 12 of these that are based on microsatellite repeats. And when we run them on a capillary electrophoresis system, you can see the size differences in these alleles to about a 2 base pair resolution. And then we'll use methods like structure to assign ticks to different populations when possible. Second, we have real-time PCR assays for resistant SNPs for synthetic pyrethroid chemicals. And I'll get to those a little later in my talk. What I will tell you right up front is the new genetic data from Rhipicephalus microplus reveal a highly dynamic invasion system in southern Texas. Let's get into some of those data. Oh, I guess first I should tell you something that is a resource we were able to make use of are tick collections made by the APHIS field inspectors. This is really amazing because there are tens of thousands of ticks that are stored, or not every year, maybe it's 1,000 to 2,000 ticks a year, but they are stored in frozen archives. And so there are ticks from cattle, ticks from white-tailed deer, sometimes from live deer that are netted from a helicopter. That is rare. Those are just focused studies. More often, we get ticks from white-tailed deer and nilgai from hunter check stations. And so we've got all these ticks that the USDA saves and stores. And then our collaborator, Dr. Pia Olofsson at the USDA, extracts DNA from the priority locations that we have questions about and sends us the DNA. So that part of it is amazing. It is wonderful to have Dr. Olofsson's lab send us ticks. She has sent us over 5,000 ticks in the past 10 years. So these are just photos showing scratching ticks from live hosts. So let's get into some data. On this map, we will talk about the genotyping with 12 microsatellite repeat loci from students and staff in our research group. And these are collections that were collected in 2017 to 2019. As you can see by the key, the ticks that come from Mexico are red triangles. Ticks from Texas are shown in these gray circles. And these ticks can be from cattle or white-tailed deer or nilgai. Most of those wildlife ticks are sampled down here near the Gulf of Mexico, but there are white-tailed deer all along here, and the white-tailed deer are both in Mexico and the US. We don't get to see what happens with the white-tailed deer as often, just because they are a wildlife host. So we've genotyped, and then we're going to take those data and try to assign them to different populations using the program structure. So here is what we found. You will see all these different colors. So structure tries to find distinct populations. And that it did that in some cases. And this is color-coded. So you can find the pink population here shown on the map. What this is, the y-axis is a graph of the probability of membership. And in this particular run and structure, we're asking the software to assign ticks to 6 different populations, theoretical populations, and it worked really well. Each tick is a very thin bar on here. And so we've got over 800 ticks showing in this run with structure. And so you'll see some of these very solid colors show to be distinct genetic groups. And as I said, you can search and find all of these on the map. But what about these other ticks in the quarantine zone in Mexico? They look like a mess. Structure cannot assign those ticks to any one group, so it's trying to assign them to multiple groups, like 3 or 4 different gene pools. And that's showing us very important information. The ticks that come from Mexico and the quarantine zone have a lot of gene flow, a lot of movement. And so they are a very diverse population. It's only the ticks that are north in the free area that become distinct. Why are they becoming distinct with this signature? It's because they're isolated. These ticks have dispersed far north of the invasion zone. And they form a temporary infestation that will last for 1 year or 2 years, but those are aggressively eradicated and snuffed out. And so these ticks that are north in the free area are not exchanging gene flow. What else did we learn from this? So there's a lot of diversity and genetic admixture in Mexico. And we see the same thing in the Texas quarantine zone because those ticks, many of those ticks have come from Mexico. The other thing that happens is wildlife are moving these ticks both ways. So that is why the quarantine zone will look so much like the ticks in Mexico. So that is bad news, that in the quarantine zone, we don't see, well, that we are seeing tick populations that look very much like what is coming across from Mexico. That means those tick populations are not as isolated. Now, this is useful information to have for managing ticks. This map is showing how dynamic the system is. Each of these distinct groups are moving independently from the quarantine zone. Another thing that we think about this is that these distinct groups are not coming directly from Mexico. They are coming from ticks that are already in Texas and then marching to the north on wildlife. And then once they get north of the quarantine zone, they can move on cattle. I should back up. I wanted to show you next, we're going to zoom in on this area of the quarantine zone to show you a place where we were able to sample ticks here from ticks that were from a Mexican cow that had strayed across. Actually, it was 3 Mexican cattle. And a month later, the cattle from that ranch in Texas became infested with ticks. And so again, triangles are ticks from Mexico. And then ticks from Texas are the white circles. When we look at this one location, we are able to run a test using an FST statistic. That is a fixation index that shows us something about genetic structure. And so FST measures genetic structure. If FST is 0, then there is no genetic structure. all the ticks have been sampled from the same population. And that is what we see here. When we compare that location to others around it, we do not see an FST of 0. We don't see very big FST changes, but none of these other comparisons with the ticks from Mexico at this location were at 0. So we were able to kind of capture an event that shows where ticks cross and then jump onto a herd of US cattle. There was another clue here that helped us understand this movement, which is that there was a rare resistance mutation that we know is in Mexico, but does not show up in Texas. And it entered this location in 2018. And it actually did make it to some of the surrounding ranches, but it was then eradicated. So this rare resistance mutation did not spread out of this. So tick movement is cryptic, and it's difficult to find a situation like this. But genetics help us to find, on a very fine scale, to find how ticks are moving. Let's go to the next slide. We had just zoomed in. Now let's zoom back out to the level of North America, because we started this work in Texas, but we really wanted to know what is the level of population genetic structure in Mexico, and would that help us understand how ticks are getting into Texas. This was our first collaboration with Juan. He and his lab group collected over 1,500 ticks from Mexico, and you can see all of these locations. I should mention that any one of these locations is a collection of about 30 ticks. And so when we do population genetics, we need a sample of that population. So there's 30 ticks at each of these. As you can see here, Juan is picking ticks off of this cow. Even though he is a respected laboratory scientist, he's not afraid to get his hands dirty. And so it was fun to visit some of the ranches. And these are data from, or these ticks came from Jalpan de Serra in Mexico. We also have one of Juan's previous students. I think this is Roberto, if I remember correctly, helping us with keeping this cow calm while we were picking ticks. So again, we can look at the FST index to see if there is structure in Mexico. And this is what we found. So in Mexico, this FST value, when you look at the average structure across all of the country in Mexico, that is low. It's only 0.03. If there's no structure, it's at 0. And so this was a surprise. I did expect to see some regional structure, but there is not much. That is a sign that there's high gene flow, lots of genetic admixture across the entire country due to frequent movement of ticks, probably from long distance transport of cattle, of ticks on cattle. When we look at other subsets of ticks, we see some interesting patterns. The ticks from Mexico that are in the quarantine zone, they look just like the rest of Mexico, which makes a lot of sense. When ticks get to the quarantine zone, there is a slight increase in genetic structure. And so the eradication program is having a slight effect on ticks in the quarantine zone, at least on genetic structure. And then in the free area, that's where we really see the genetic structure increasing. And that is good news. That is telling us that the eradication program does have an effect. If it wasn't having an impact, there would be ticks all through southern Texas, and we would see FST drop. So this is a tool that could be used for monitoring. And you might imagine that you could use that in some of the eradication zones, say in Mexico, where these yellow areas are where the eradication program is acting. And if there were ticks that escape, into a yellow zone, we could monitor for that population structure to see, is an infestation isolated? It would need to be compared to ticks from nearby, but it could be something for long-term monitoring. Good. And so let's move into the resistance markers now that we use for population genetics. So the model for resistance with synthetic pyrethroids is very well known. It is in the voltage-gated sodium channel, and there are these mutations that occur in the transmembrane regions, so that these target site mutations prevent the chemical from keeping that sodium channel open, and they lead to resistance. And so our work at Northern Arizona University has shown that in Mexico and North America, we have all three of the known SNPs for ticks. And originally we thought there was only one of these mutations in Mexico, but we have found all three of them. So when you know about these genotypes, you can predict the phenotype. So the y-axis is the percentage survivorship. And of course, that goes up as you get more and more ticks that have resistant SNPs. Especially if a tick has two resistant SNPs, then it seems to be more resistant than the heterozygotes that just have one. So we wanted to see what where the genotypes are in Mexico. And again, we used the 2016 samples that Juan shared with us. And here's what we found, that we have a lot of locations where over 75% of the ticks have resistant mutations. That's all of the red dots. The orange dots are also bad news because those are expected to be highly resistant ticks. There were some surprises. There are completely susceptible populations at some locations along the Pacific side of Mexico. And in locations that we found this, synthetic pyrethroids could still be useful as an acaricide, but the ranchers would have to know that. Otherwise, you could end up with applying more synthetic pyrethroids to a population that's already resistant. And then you can see in Texas, in the past, we used to have mostly susceptible populations. But in more recent years, we're at about 50% of the tick collections are resistant. So in this map, again, these are the same collections I showed you before for the population genetics study from 2017 to 2019. The susceptible are shown in blue. And if it doesn't have blue, then these are all resistant populations. Some are more resistant than others. This area has a very high level of resistance, whereas ticks farther to the west, they have resistance mutations, but the population is still somewhat susceptible. But the blue are completely susceptible. You can see there's a concentration of susceptibility here to the west, and also some in the ticks that are right along the border. And so you can see that we did have a case of resistant ticks making it far to the north, being spread on a cattle shipment that came from closer to the border. The other issue is that in this area with all the wildlife, we see a lot of ticks that are resistant on white-tailed deer and nilgai. So they can easily spread ticks that are resistant into fields that have cattle. So I'll cover some main points here that we have management implications that we have learned from these population genetic data. First, it's really great to have the USDA frozen collections. It's a highly valuable resource. And when we genotype all those ticks, we find that short distance tick movements are common, and they often happen on wildlife hosts and stray cattle, and also domestic cattle. Long distance movements, definitely that is happening on cattle. It's ticks, it's cattle that have not been monitored before the shipment. And usually it happens when the cattle originate in the free area to the north of the quarantine zone and then are moved long distances. Resistant ticks are spreading throughout Mexico, and we do find them entering Texas. And that it's important to screen for resistance. We wouldn't have known that without using genetic tools to screen a lot of these. The USDA does a lot of larval packet testing to monitor for resistance, but they are not able to do that with every collection. Whereas with DNA-based methods, we can screen many more ticks. And then this new signature that we have identified, that you can look at population genetics to see if your genetic structure rises. That's an indication of isolation, and that's a good sign for eradication. But given this current level of movement of tick and cattle that carry Babesia, there is still a threat of reintroducing the disease. Because of that threat, we have also developed other tools to look at these tick populations. As I've been explaining to you, we are very focused on analyzing the tick populations. We do not have access to the cow blood or to look for pathogens. So we've developed some methods to screen the ticks to see if we can find the parasites and bacterial pathogens. So we've got different genes that we use to detect Babesia bovis and bigemina. We are also looking for Theileria equi and Hanei, which are horse pathogens. Because we're looking at 18S, we sometimes are finding a native deer pathogen, which is Theileria cervi, and then we screen for Marginelli. We're using the Illumina MySeq platform, and that generates short reads. So all of our targets are very short, less than 250 bases. When we did this, again, using this great resource of ticks from Mexico, we found Babesia bovis at a couple of sites far to the south and in Zacatecas. And so we just found 2 locations. Bigemina, we did find in the yellow circles, we found in 5 locations. So we didn't find a lot of Babesia, but then our samples were not focused on an acute outbreak. And so we probably would have found a lot more if we knew that there were cattle that were experiencing this disease. However, with Anaplasma marginelli, we did find it everywhere. It was over 90% of the locations had at least a few ticks with Anaplasma marginelli. That's all shown in purple. In Texas, we didn't find any Babesia. We did find Anaplasma at 3 locations. It turned out that 2 of those were from Mexican stray cattle, but one of those collections was on a Texas cow. And so the Anaplasma, there was a risk there of Texas cattle contracting that disease. Interestingly, we did not find Anaplasma marginelli on ticks or nilgai, on nilgai or white-tailed deer in Texas. And we had expected some of those to be positive. In addition to Microplus, we are also studying the other cattle fever tick, Rhipicephalus annulatus. And with the pathogens there, we found some interesting things. We did not find any Babesia bovis or bigemina. We screened 676 ticks. So we had many years to look at pathogens. What we did find was Theileria equi and Hanei, which are horse parasites. And these are known to occur in Mexico and southern Texas. Now, the US is considered free of these parasites, but occasionally we do get outbreaks of them. But the really surprising part, so, we might expect to find Theileria positive ticks on horses from Texas and Mexico, but we were finding the positive ticks on cattle and white-tailed deer and red deer on these other hosts. So that suggests to us that the ticks must be feeding on a horse first to pick up the Theileria, because Theileria is not transovarially transmitted. Those ticks must leave the horse and then seek other hosts to try to find a cow. And sometimes they find cattle, but other times they find deer. Okay, so I should move along here. I've got just a few more slides. This just shows that with the 18S sequences, we can sometimes get some genotyping capacity with the amplicon sequencing technique as well. And so in the red are the new sequences that we found in Rhipicephalus annulatus. We have our next research move is funded. It has been funded for 3 years. We're just wrapping up this project. And here is the team that is participating in this project. And we brought in Dr. Guy Klafke as well for this one. And the two main questions we have are to look at RNA sequencing from lab colonies that are resistant and susceptible, to learn more about the mechanisms of resistance to these chemicals. We don't know the mechanism as well as we do for synthetic pyrethroids. And so there's a lot of room for research in this field to find out what genes are being expressed that lead to this resistance. And Guy worked very hard to work on getting us RNA from resistant and susceptible lab colonies. We are still analyzing those data. We do have the RNA-seq data now, but I'm not going to present that today. I am going to talk to you about the second main goal of this project, which was to characterize diversity at genes being considered for anti-tick vaccines. And so we've heard a little bit about anti-tick vaccines this morning, and Dr. Fish gave us a very good example of how to develop the epitopes that would be efficacious against ticks. We are not looking at salivary proteins. We're looking at a list of others. And of course, the existing commercially available vaccine is based on protein VM86. We are looking at diversity, and we're doing this by using DNA and using amplicon sequencing to, let's see, did I skip a slide here? Well, so we're using amplicon sequencing to look at exons. Remember that eukaryotes have these huge introns, and this can be kind of a pain to sequence. I did skip a slide. So we are going to investigate 11 genes that have different biological functions. So trying to get at gut transporters and pathway signaling, water metabolism. We have one target that is part of the salivary gland apparatus. That's a metalloprotease 4. And then other interesting targets will be reproductive targets like egg storage proteins. And we are looking at diversity there because these ticks have a lot of diversity in their genome. And the peptide used for vaccination might not be fully protective if it can escape. So we're using amplicon sequencing to sequence all of these exons. These green arrows represent primers that we can all put into a single PCR and then amplify everything at once. Our samples, again, are coming from Mexico, Texas, but other countries as well, Puerto Rico, Colombia, and Brazil. And here are the genes that we have data for. We don't have a lot of sequences for most of these yet, but we do for BM86 and MP4. We're also looking at multiple species of ticks to try to see if a vaccine could be used for more of the Rhipicephalus species. So here's just a broad overview of what we found, a lot of genetic variation in BM86 and also in metalloprotease 4. This is just showing the number of SNPs on the y-axis per exon. Some of these exons have a lot of diversity. Others don't have too many SNPs and show a better degree of conservation. When we did this, we also used what is available in GenBank to look at these. So there were 184 sequences available, and none of these matched the protein sequence for the commercially available vaccine. When we look at this in terms of genetic distance, instead of looking at exons, we can see BM86 has the highest level, but part of that is because we're looking at several species of Rhipicephalus, and so this blue part really is a large part of the bar. But within Rhipicephalus microplus, you can use the gray and the gold bar to kind of see which genes have a little more conservation, and things like VDAC and subolecin, GST seem to be more conserved. We still have to sequence many more ticks before we can be sure of the level of conservation. And one last slide is just to show a case example of what we can do here. The Petarroyo et al. Publication shows 3 peptides from BM86 that are being developed into a vaccine. And these are in exons 2, 4, and 11. When we look at diversity in these specific exons, we do see some diversity shown by the pink box. Those are polymorphisms that we see in the ticks from North America. I don't know if these same polymorphisms are important in Brazil, which is where the new tripeptide vaccine will be used. But you can see that there are examples of diversity. These peptides are not fully conserved, but it's hard to know if a single amino acid is enough to prevent the vaccine from working. With that, I will close and acknowledge a really great team. I very much appreciate working with these scientists, and I appreciate the funding from USDA. And thank you for inviting me to present to the meeting today. I will stop there.
Thank you very much, Joe. Great presentation, very informative. Actually, Patricia Cervantes Acosta says, "Good morning. I consider this information very important for the development of local research. This will allow current and specific knowledge for the different geographical areas in Veracruz state in Mexico." And Dr. Carlos Vega is asking, cattle tick inspection in the Mexico-US border is very stringent. Do you believe that mixed populations come from domestic or wild animals or both?
Thank you for asking that question. I neglected to really focus on the answer, which is, I think the mixed populations are moving across the border on wildlife. But the genetic diversity, I think, is getting there on cattle that are transported up to the northern Mexico. And then those ticks on cattle will use whatever host is available, and they can successfully use white-tailed deer. And so that is the source of the ticks moving across their river. When we import cattle from Mexico, the ticks have been cleaned off. There can be resistant ticks on those cattle, But we really don't see cattle fever ticks on those imported cattle once they're in Texas. We just don't get reports of that. So it seems that those cattle have been very much cleaned up.
Mm-hmm. Okay, and also Dr. Vega has another question. Do ticks from different populations, say cattle or wildlife, mate? Have you measured such event?
Yes, we have. Thank you for that question, Dr. Vega. And we have a publication on this from 2014 where we have deer from one pasture where we also have cattle, and we looked at the ticks in the deer and the cattle, and there was no difference. So this FST was at 0, so there was no genetic structure. So we know that the ticks can move back and forth from wildlife to cattle, from cattle to wildlife. There is nothing preventing the ticks from exchanging gene flow between hosts.
Mm-hmm. Okay. And I have one last question, Joe. I was seeing your results on the pathogens that this resistant tick have. Do you know there is a way to know whether resistance has an effect on pathogen survival in the tick? Is there a way to measure or to know that?
That would be great to know that. And so the resistance, as long as the chemicals are being used and the resistant ticks are more successful, it seems that at least some ticks will be able to be competent vectors for Babesia. But we did not find, so we have tested that. Glenn Scholes looked at that. We did not seem to see much of a difference in vector competency in a resistant population that he had tested. There might be other factors that would lead to a population of ticks that are better as vectors. But we still have a lot of work to do there to find that out. Massaro alluded to that. There are many things still to find out about Babesia and interacting with ticks.
Mm-hmm. All right. Well, thank you very much. I appreciate really you participating in our symposium. It was a great presentation. Thank you.
Thank you, Juan.
And before we move to the next speaker, I would like to share with you the video of our the company that is helping us to promote this symposium, Elanco. Adelante con el video de Elanco.
Elanco acaba de expandir sus horizontes. Ahora, toda la línea de productos de salud animal de Bayer, reconocida mundialmente por su alta tecnología y calidad, hace parte de nuestro portafolio y llevará a nuestra marca por nuevos caminos. Es así, expandiendo su participación y seguimiento, que Elanco consolida su liderazgo e innovación en la prevención y tratamiento de animales de producción y animales domésticos. Bienvenido al movimiento Elanco de crecimiento saludable de las relaciones de negocios, pero lo más importante, de la salud y el bienestar animal. Elanco, porque crecer es saludable.
All right. Our next speaker is Dr. Susan No. Is she around? Hello, Susan.
Hi, Juan.
Nice to see you.
You too.
I don't know if you can share your presentation now, just to see if everything is working.
I think I am. No? Here we go.
All right. Okay. Then I'm going to present Dr. Susan No. Dr. Susan No got her DVM degree at Washington State University in 1996. She got her PhD at the same university in 2007. And she's a research scientist with the United States Department of Agriculture, the Research Service, the Animal Research Service at the Animal Disease Research Unit in Pullman, Washington. She is an adjunct faculty at the Paul Allen School of Global Animal Health and the Department of Veterinary Microbiology and Pathology at Washington State University. She's also a diplomat of the American College of Veterinary Pathologists. Her research goals are to develop methods to prevent tick-borne diseases. Her research efforts are focused on principally Anaplasma marginale at both the vector pathogen interface and the interface between the pathogen and the mammalian host. Susan, thank you for participating in our symposium. So you can go ahead and give us your talk.
Thank you, Juan. I think the thing you failed to mention is that you actually taught me how to use pipettes as well. So it's really, really nice to be here. And I'm going to share a bit of our ongoing research. These are works in progress about work we're trying to do to identify adhesins of Anaplasma marginale and the mechanisms that Anaplasma uses to get into tick cells. All right, so Anaplasma marginale is, of course, one of the most common tick-borne pathogens of cattle worldwide. It causes bovine anaplasmosis, and that disease is typified by anemia with production loss and often outbreaks of death. And it's transmitted by exotic ticks. So this pathogen, it's important for these studies for me to really emphasize that this is an obligate intracellular bacteria. And as such, it has to actually get into the cells and replicate, or it dies. And then it must, of course, transition between the mammalian host, the red blood cells, and the tick. So this is a histologic section stained with hematoxin and eosin of a dermocenter andersonite tick. And that's our tick vector that's most relevant to my region. And this is the anterior end of the tick, and here's the posterior end. Here's a little bit of remnant cuticle. And all of these globular bits here are the salivary glands. And here's some of the midgut. So, the long-term host is the bovine. And so here, this is a red blood smear, or this is a blood smear stained with Giemsa, and you can see all of these little colonies of Anaplasma, these blue dots in the red blood cells. So when the tick ingests the blood, Anaplasma then has to, leave the red blood cells, which are lysed in the lumen of the midgut, and enter these cells and create colonies. And that's what these red-brown blobs are. These are colonies of Anaplasma. The Anaplasma then transits to the salivary glands, where again it has to enter those cells and replicate. And then when the animal takes another blood meal, the foundation of this work really is that knowledge gaps in our understanding of this interface between the tick and the pathogen and the host and the pathogen. Those knowledge gaps really limit our ability to prevent bovine anaplasmosis. Now we know some about what Anaplasma does once it's inside the cell. So this is a cartoon of a tick midgut cell, epithelial cell. And Anaplasma, it gets into this cell and it establishes this nice niche, and it controls this niche. And it's aberrant compared to the other vacuoles in the cell. It's labeled with markers for early endosomes with Rab5. It's labeled with markers for a recycling endosome, which are Rab4 and 11. It's labeled with Rab7, which is for a late endosome. And importantly, this vacuole never becomes acidified, and it's often adjacent to portions of rough endoplasmic reticulum, which allows nutrient acquisition. And the Anaplasma control this vacuole through some sort of protein production, and they replicate quite happily here. If you treat these cells with tetracycline, the vacuole, which damages the ability of Anaplasma to make proteins, the vacuole becomes acidified and the anaplasma are killed. And this is an electron photomicrograph of one of these colonies of anaplasma inside a cultured tick cell with a little bit of endoplasmic reticulum. So we know a little bit about what happens inside the cell, kind of the basics, but we really don't know how it is that anaplasma actually enter those cells. We don't know the molecules that are used for the most part, and we don't know the mechanisms that are used. Okay. And some of this that drives this knowledge gap is really the fact that we know there's some real knowledge gaps in tick physiology. And tick physiology is unique compared to other invertebrates. My favorite example of this is about the tick midgut. And so, as many of you may know, of course, the blood meal is lysed within the midgut, and this moth-eaten looking red blood cell represents a lysed red blood cell. And then the blood meal is taken up by these midgut cells, and it's actually digested inside the cells rather than in the lumen. So these are highly phagocytic, highly active cells, and all this intracellular digestion. It's also a very coordinated process. For example, hemoglobin is taken up in a receptor-mediated process, which is illustrated by these green little receptors here. And so I think that hemoglobin is taken up by receptor-mediated endocytosis, and albumin and other blood meal components are probably taken up by a less specific process, by macropinocytosis. And all this to really say that intracellular pathogens and anaplasma as well exploit these normal physiological processes to enter midgut cells or whatever cell, avoid digestion, and establish that replicative niche. And so thinking that the bacteria are really taking advantage of ongoing physiological processes, let me just walk you through 3 big categories of how cells take up extracellular materials in general terms. So the first is macropinocytosis. And macropinocytosis is also called the big gulp. And that's just the cells engulfing the extracellular milieu. So this has a particular morphology on electron microscopy, in that the outer membrane makes these big ruffles, and they engulf the cargo, and it becomes a vacuole that's intracellular. The next, probably best studied and best understood mechanism is kind of the canonical receptor-mediated endocytosis, and that's mediated by clathrin. And so the cargo-bearing receptors are recognized by the adapter proteins, which are these orange bars. And then that triggers clathrin to assemble into a lattice-like network. And the clathrin here is depicted by these little green triskelion things. And so then this cargo is taken up into the cell and trafficked wherever it needs to go. And this also has a particular morphology on electron microscopy, and that you can actually visualize these clathrin-coated pits of these electron-dense areas. And finally, there's lipid raft-associated endocytosis, and this is probably the least well understood and studied. And so this can be with caveolin. And so caveolin is, again, a membrane protein that associates very tightly with lipid rafts. And it's depicted here in purple. And again, you've got your cargo. It's taken up into this caveolin line pit into the endosomal system. There's also uptake systems that are associated with lipid rafts, but don't require caveolin. And if you look across pathogens and viral pathogens and intracellular pathogens, all of them use a variety of mechanisms to enter cells, and all of these different pathways are exploited by these different pathogens to enter cells. And this has to do with the cell type of entry and the pathogen. But really, when we think about tick-borne bacterial pathogens, there's no data out there, at least that I've been able to find, that really talk about the mechanisms that pathogens exploit to actually get into the cells, and particularly not in anaplasma marginality. So what we've started to do is to use chemical inhibitors to block these pathways in order to measure, and then measure pathogen entry, to try to figure out what mechanisms the bacteria is exploiting to get into the extracellular niche. And we're using inhibition assays to do this. And so what these look like, we take our dermocenter endosomni cells, and these are DAE100 cells, and these are an embryonic cell derived from dermocenter endosomni. And so as such, as a cell culture, you know, there are some limitations. That's also a very practical system that gives us a nice first start. So we put those cells into wells. We treat them with inhibitor for 30 minutes. And then we infect with Anaplasma marginale with about 100 bacteria per cell for 1 hour. We replace the media and we add gentamicin. So the Anaplasma have about an hour to get into the cells, and then we add the gentamicin, and that should kill the extracellular bacteria. We change the media, and then we harvest the cells through time, 24, 48, and 72 hours. And because we only want to measure the live Anaplasma, we isolate RNA and do reverse transcription and real-time PCR. And then we calculate the Anaplasma levels relative to the tick cell numbers. So the first two, we've started by asking the question, you know, does clathrin-mediated endocytosis play a role in Anaplasma marginale entry into these tick cells? And here's the cartoon to remind you what that looks like. And we've used two inhibitors. The first is chlorpromazine, and that sequesters clathrin. And it also inhibits the endocytic recycling of clathrin. And then we've used monodensyl cadaverine, MDC, and that works in a different way, and it prevents the assembly of clathrin-coated pits. So all of these graphs are set up the same. And along the vertical axis is the log base 2 relative expression of Anaplasma marginale. On the horizontal axis are the hours post-infection. Now the control are cells that are treated with the carrier chemical. So these inhibitors are diluted either in DMSO, ethanol, or water. And so the control has whatever the appropriate carrier is. And then of course there's the treatment, which is the drug of interest. So with chlorpromazine, we start to see, you know, at 24 hours, we start to see possibly a decrease in the amount of Anaplasma in the cells. And this is 24 hours after the cells were infected. This is statistically significant, but I kind of raise a little eyebrow about that. And then at 48 hours, the change is much more clear in that we have less Anaplasma, you know, they're recovering from the cells after they've had some time to grow. And then at later time points, the Anaplasma seem to start recovering, they start to grow, and again, with our control, they're also starting to grow. Now with MDC, we don't see the same phenomenon. Really, at 48 hours, there's really no difference between the treatment and the control. And the same for 72 hours. And so this really suggests that I really think that clathrin-mediated endocytosis has a prominent role in Anaplasma entry into tick cells. And we'll talk a little bit about some of the limitations here a bit later. And then for cholesterol-dependent endocytosis, again, we're using two chemicals. One is methyl beta cyclodextrin, and that sequesters cholesterol from the membrane, and philippin-3, and that's supposed to be more specific for inhibiting calveoli-mediated uptake, and that it impairs this membrane invagination of calveoli. So with methyl beta cyclodextrin, we start to see a decrease in the amount of Anaplasma we can recover at 24 hours, and a bigger decrease, a bigger difference between the treated and control at 48 hours. But with philippin, there's really no difference at these two time points between the treated and the controls. So I think this suggests, particularly at this 24-hour time point, that maybe lipid grafts have a role in Anaplasma entry, although it's frankly quite a small difference there. And then we get into the issue of how these data, there's a little bit of confounding here because Anaplasma have to establish that niche. And so if you look here, the numbers are starting to decrease. So that would suggest to me that the anaplasma, they maybe didn't get in as much, but they're also possibly not getting the nutrients they need, particularly when the numbers start to decline rather than recover and go up. And we know from nice studies done with Anaplasma phagocytophilum that cholesterol is required for anaplasma growth, and the bacteria acquire that from the host cell. So my suspicion here is that while cholesterol may have a role in anaplasma entry, it probably also has a really important role in the growth of the bacteria. And so in this case, the anaplasma may not be able to get that cholesterol, and thus, they're dying through time. At least they're not replicating. And with Philippin, again, I think at this later time point, that also may suggest that there's some disruption of the ability of the bacteria to get nutrients at these later time points, but that calveoli really don't play much of a role in anaplasma entry into Tec cells, at least in this system. So for macropinocytosis, and this is the one I was really curious about, because this is just that big gulp mechanism where the cell is just taking up the extracellular milieu. And if anaplasma enters by that route, it's going to be really hard to find ways to block that process in order to block eventually tick transmission. So we use two inhibitors for this. One is EIPA, and that inhibits NAH exchange. And the result of that really is that you alter the charge of the inner plasma membrane. And then we used imipramine, which inhibits that membrane ruffle. So with these two inhibitors, at 24 hours, the findings are really quite similar in that we get a marginal decrease. They're not statistically significant in the amount of anaplasma that gets into those cells. And then at 48 hours, the anaplasma recovered. And they continue to grow, and the cells were treated with EIPA, but not so much with the imipramine. And to my mind, I think this suggests that the anaplasma do, to some degree, enter by macropinocytosis, but those levels recovering at 48 hours makes me wonder if the anaplasma getting in through this route is an aberrant pathway for it, and they don't survive. And so what we're seeing here are the anaplasma that have actually entered through a different mechanism, through receptor-mediated endocytosis. Okay, so a bit of a summary here. And at this point, it's possible that anaplasma gets into tick cells through multiple routes, although from these data, it looks like the clathrin-dependent endocytosis may be one of the more prominent routes. And it's possible that cholesterol, but not calveolin, mediates some entry. And that macropinocytosis likely represents an aberrant entry pathway. But like I said, we need, we need some additional studies to really verify these data. And I propose we're going to use a combination of inhibitors. And then one of the problems here, and I think one of the reasons you see the discordant results, particularly with CPZ and MDC, is that the specificity of the inhibitors may be a problem. And so I think we need to use another method to confirm these results. I would propose to use RNAi. It would be interesting to do this in some other tick cell lines. And then ultimately, we need to confirm this in actual tick tissues. But like I said, I think it's really encouraging that macropinocytosis is not much of an entry route. And I think this allows us to think about really targeting that adhesin receptor interaction as a means to disrupt tick transmission. And then if you look at this in the broader context, if we could find all of those adhesins that mediate entry of bacteria both into the mammalian cell and into the tick cell, this would broaden our tools to develop an immune response against these molecules to disrupt this essential function and build a more robust vaccine to prevent bovine anaplasmosis. So we know some about the anaplasma marginality adhesins. MSP1A, it's major surface protein 1A. This is a little graphic of it. It has an N-terminal, a graphic of the predicted transmembrane domains. So there's the N-terminal repeats. This is often used for genotyping. This portion is thought to be extracellular. And these N-terminal repeats, there's nice data that suggests that this mediates adhesion both to tick cells and red blood cells. And then the C-terminal portion has these 4 transmembrane domains that are predicted. Now, the story here gets to be a little bit more complicated. There's a lot of strain variation with Anaplasma marginale, both genotypically and phenotypically. And so an example of this was done in, this work was done in Masaru Ueti's lab, but the Mississippi strain is a strain of Anaplasma that doesn't really enter or replicate in the dermocenter Andersoni midgut. And so that's illustrated here in that he tested these 3 Anaplasma strains. The St. Mary strain is our prototypical research strain. It's very tick transmissible. It causes, it's a virulent strain. And then he compared that with the Mississippi strain and the Anaplasma marginale or Anaplasma centrale. And these numbers are from dermocenter that were infected as adults and then have now transmission fed. And so the St. Mary strain, we typically see about 10 to the 6th, 10 to the 7th bacteria per midgut, which is what you saw here. The Mississippi strain, there were just no bacteria in there, right? So the Mississippi strain is unable to actually either enter or replicate in the midgut. And yet it has these tandem repeats of its MSP1A that are identical to transmission competent strains. And additionally with Anaplasma centrale, it can replicate. It also is poorly transmissible by dermocenter Andersoni, but it can enter and replicate in the tick midgut up to about 10 to the 5 copies per midgut. And yet it actually lacks tandem repeats. And so I think the story with MSP1A as an adhesion is a complicated one. And then MSP1B, there's nice evidence from the De La Fuente lab that it's an adhesion for red blood cells. Now, the other known adhesion for Anaplasma marginale is OMP-A. And this work was done in Jason Carlion's lab. And here's a schematic of how this is done. So this is a host cell, the nucleus, and a little blue receptor here. And then the Anaplasma are represented here in blue ovals, and this yellow dot is a really extra big surface protein that serves as an adhesion. And then there's a recombinant adhesion. And so in a cell culture system, you've got your cells, so it's a tick cell, you've got your little receptor, the Anaplasma bind this receptor, they get into the cell and they are able to replicate in their vacuole. So in order to test whether this recombinant protein could serve as an adhesion, you do a competition assay. So you mix this recombinant protein with your Anaplasma marginale, and then that will, those recombinant proteins, if they bind this receptor, it will prevent the Anaplasma or will reduce the amount of Anaplasma that can get into the cell. So they did this with Ixodes scapularis cells. Ixodes scapularis is not a vector of Anaplasma marginale, but these tick cells are very, very permissible to Anaplasma infection. And they also did this with endothelial cells. And they saw a moderate reduction in the percent of infected host cells with Anaplasma, about a little bit more than a 25% reduction in the case of Ixodes scapularis, and about a 25% reduction in the case of the endothelial cells. And then these other panels, they enumerated the number of Anaplasma vacuoles per cell. And again, with competition with the recombinant Anaplasma, with the recombinant OMP-A, they got a decrease in the amount of Anaplasma that could get into the cells. The control in this case was the recombinant tag, the GST recombinant tag on this protein. So altogether, it looks like we know that OMP-A is very likely an Anaplasma marginale adhesion. This has not been confirmed in red blood cells or a relevant tick vector. There's good evidence that both MSP1A and MSP1B are also adhesins. And because this is such an essential function, I think there are likely others. I think this is probably a highly redundant process. So what we've set out to do is to identify the full array of anaplasma marginality adhesins. Both for so what are those surface proteins that mediate adhesion and entry to the red blood cells and to tick cells? And we did this using a phage display library as the first screening tool. So phage are viruses that infect bacteria. And we used a T7 phage that will express exogenous DNA as protein on its surface coat. And so we constructed a library composed of anaplasma marginality outer membrane proteins and all the vaccine candidates. We clone those into this phage, into this T7 phage, and then all of these candidates are expressed on the surface coat of these viruses. And so we have our library, and what we ended up with were 81. We tried to insert 81 different genes. 10 of them we just couldn't clone in. And then we would use real-time PCR to measure the input phage population, so we know what's going in. And then we mix this phage display library either with our dermocenter cells or with our red blood cells. And then you wash and you remove the unbound phage and you recover the bound phage. And then you do this through repeated cycles. It's called biopanning. And at the end of it, you recover the phage that were bound to your target cells. So these are the phage represented here and here. So these were the ones that were actually bound to the target cells. And to recover them, you have to culture them. And then again, we used real-time PCR to identify the specific, insert that's in that phage and get a rough idea of quantitation. And this whole system was set up by Deb Alpern and Jesse Usko. And it was a ton of work. So, like I said, phage display, I think it's best used as a screening tool because there are some real biases to it. For example, the insert size. So we put in different lengths of DNA into our phage. And so here's our number of base pairs per insert, and then here's the percent of those inserts with that particular size that were recovered. And this was for the DAE cell recovery and for the red blood cell recovery. And so what you can appreciate from this is once your phage, once the inserts got above about 1,500 base pairs, you're much less likely to recover them. And so is this a function of, long genes are not, adhesins, or is this a function of, the phage really just don't like having these big inserts in them? And it's probably the latter, or maybe a combination. The other bias that really comes into this, because we have to grow those phage in order to recover them, is that some of them will replicate better than others. And so we're much more likely to recover those phage that replicate better. So that said, let me show you what the data look like, what our results are. I have a number of tables here, and I've broken them out by kind of category of protein. So in the first column is the gene name. And then these are the DAE cells. So this is recovery from DAE cells, recovery from red blood cells. And then the empty column is super important because phage some of them will just stick to plastic non-specifically. And so that's really our control. So if we had a phage that would always be recovered from the plastic, would always stick to the plastic, we discarded that and essentially said that we didn't test it, because if it always sticks to this plastic well, we don't know if it's sticking to the host cells or not. And then the percentages. So we did this between 6 and 9 times, depending on the cell, depending on those red blood cells or DAE cells. And so what this percent represents is just a plus minus. So say, for example, with the 50%. So in 50% of our replicates, we recovered the phage that was carrying the TOL-C gene. And then the number in parentheses are the CT values. So to give a rough estimate of the amount of phage that was recovered. And remember, a high CT value means that there's not many genome copies there. So lower CT values equate with more abundant genome copies. Okay, things complicated. And finally, we get to the color coding over here. And so to kind of highlight which are the best and second best candidates, I highlighted them in green and yellow. And so the green, this is just based on the percentages. So the green ones are those that were covered 3 times more frequently in a target cell than from the empty well. Right. So for example, with OMP13, we never recovered it from the target cell. We recovered it 50% or from the empty well. We recovered it 50% of the time from the red blood cells. And so 50% is 3 times greater than 0. And so it's a good candidate. It's also one of the few that was really a better adhesion in red blood cells than DAE cells. So now the other gene here that's really important is OMP-A. So OMP-A, there's evidence it's an adhesion. And in our studies, we did recover it some from the empty wells, but we recovered 100% of the time, both from the red blood cells and the DAE cells, with reasonably high CT, with reasonably low CT values or a reasonably abundant amount of the phage. And so this gives us some evidence that this is there's some specificity to the system. And then also a lot of these we never recovered. So all of these different genes were present in the input population. They were never recovered from the empty wells or from the red blood cells or the DAE cells. So we're losing a lot of our candidates because they're not adhering. Again, giving us some confidence in the assay. All right. So the type 4 secretion system, again, this is the system that is expressed by anaplasma marginale, and it injects anaplasma proteins into the host cell to help to control that cell. There's many components of the type 4 secretion system that are vaccine candidates, and the ones that we found as potential adhesins were fairly interesting. So VIRB3 was our best candidate. VIRB3 is part of the surface pilus. It sits right here on this schematic. So it's exposed to the outer membrane. This is the host cell membrane here. This is the bacterial membrane here. So it's potentially exposed to the outer membrane of the bacteria, and thus potentially exposed to the immune system. And it would make sense that it would adhere to things. It's part of its job. And then we also detected these kind of moderate candidates, VIRB11, which is an ATPase, and so that doesn't make sense. So I suspect there are, of course, some false positives here. And then VIRB9 and VIRB7, they are part of this pink complex. So again, they're part of the outer membrane components of this secretion system. Now, the major surface proteins, these proteins are just the best known. They're immunodominant. They've been studied the longest. The only one of these that was much of a candidate was MSP3. This was a bit of a surprise. We recovered it 100% of the time from DAE cells and 83% of the time from red blood cells. And this is thought to be, well, this is involved in antigenic variation. And then the family that had the most hits was the MSP1 multi-gene family. And we're going to talk, and you can see here that many members of this multi-gene family we recovered as being quite good adhesins to the DAE cells, as well as the erythrocytes. Unfortunately, MSP1A was not included in the phage library. We couldn't actually get it cloned in. We're going to test this anyway in downstream experiments. And then there's all of our hypothetical proteins. And again, most of these we didn't ever recover from the DAE cells or the red blood cells. The exception being AM197 and 779. And we don't really know what they do. Okay, so here's just a summary of our best candidates. Again, OMP-A came up, which gives us some confidence in the assay. And then I want to talk more about the MSP1 multigene family. Okay, so MSP1, it's expressed on the surface of Anaplasma marginale. And it's expressed as a covalently linked heterodimer of MSP1A and MSP1B1. And in experiments done some years ago, these MSP1 protein complexes were affinity purified, so they're native proteins, and they were used to immunize animals, and they induced quite nice protective immunity. And so there again, they're very solid vaccine candidates for this reason and others. And as I said, MSP1A is a single copy gene. Now the MLPs have caught my attention, and the MLPs are the MSP1A-like proteins. And we recovered MLP 3 and 4 as really quite robust candidates as adhesins. And MLP2, we can't really talk much about because it adhered really well to the plastic wells. And if you take these, so these MSP1A and MSP1A-like proteins, if you align them, which is here, you get a 30 to 37% identity in that C-terminal end. So the MLP-like proteins don't have the tandem repeats. They don't have that part of the gene like MSP1A does. But they have some degree of identity in the C-terminal end. And we also know that thanks to Roberta Koku, a PhD candidate in my lab, that these are all expressed as mRNA. So, and we also suspect they're expressed as protein, but again, we have yet to determine that for sure. I could say though, most genes in anaplasma, if they have a promoter, they're expressed. So here's with MSP1A, here's the transmembrane helix prediction. Again, here is the N-terminal repeats, and then it has these 4 transmembrane helices predicted. And MLP2, 3, and 4 have that same structure as MSP1 C-terminus. So what we have here is some sequence variation, but some structural conservation. And to me, that really sounds a lot like it would be advantageous for adhesins because you want those to not be recognized by the immune system. You want redundancy, and yet you're going to need some form of structural conservation. But I am wildly speculating. And then for MSP1B, so MSP1B is a multi-gene family, and unlike the MSP1A family, there's a lot of sequence identity among the gene families at the protein level. So MSP1B1 and MSP1B2 are 90% identical. You can find transcript for MSP1B1 and MSP1B2, but really we've only ever found protein for MSP1B1 as part of this MSP1 complex. So probably what we're identifying here are some of the conserved regions that are present in all of these, partial gene family, a lot of these partial genes in this gene family. So this may really represent just one candidate rather than a bunch of different candidates. So what's next? So this is, again, kind of a work in progress, and we're going to, we're working on, and we have some of these candidates expressed as proteins, and we're going to go ahead and do these adhesin assays, these entry competition assays, to try to confirm or to determine which of these proteins really do mediate anaplasma marginality adhesion to their host cells. And then we hope to identify their binding domains and ultimately test in immunization and challenge trials. Okay. So we had a lot of help. I have a really great lab. Elizabeth Hoffman is a new scientist in the lab, and she's kind of taken over this work. And Shelby Jarvis is our laboratory manager, and Roberta Koku has been working on some other work with genetic variation and how that accumulates in animals through time. But with that, I appreciate your attention, and I don't know if there's time for questions. Let's see what Warren says.
Thank you, Susan. Very, very interesting presentation. Very good information. There are some questions here. I'm going to try to read them. Dr. Carlos Vega says, do you use a cell line or primary cultures?
Yeah, is that it?
Yeah, that's the first question.
Yeah, so for all of these experiments, we have used cell lines. We've used these dermocenter Andersoni cell lines. I would like to shift to, primary cultures we haven't set up, although Massaro's lab has made a lot of progress toward that end. And that's a real that's a real limitation in that at least these dermocenter cells are there's a lot of membrane turnover and they're just inherently very phagocytic. But we also, will ultimately be able to, pull out tick midguts and salivary glands, and they will live for, a week to 2 weeks in culture without the rest of the tick. So we do have the opportunity to take this work into, more relevant tissues.
Mm-hmm. There is another comment by Dr. Carlos Vega. He says, very interesting talk, Dr. No. That might explain why there are no in vitro culture of Anaplasma marginale. What do you think?
Well, we can grow Anaplasma very nicely in ISC6 cells, in DAE cells. There's, I believe, a Boaphyllus, a Rhipicephalus cell line as well. It also grows nicely in endothelial cells. Whether or not that's relevant in the cow is still an open question. And so we have these very nice tools to grow Anaplasma in vitro. We cannot, and maybe this is what you're referring to, we cannot figure out how to get the Anaplasma to actually invade red blood cells. Although a colleague of mine, and we might try this, has suggested if you could, grow those cells in the endothelial tissues, in the endothelial cell line, and then overlay them with red blood cells, you might be able to get that replication cycle such that those bacteria would come out of the endothelial cells into the red blood cells. Hopefully we'll get a chance to try that, because not being able to work in red blood cells is really a challenge, as you can imagine.
Mm-hmm. And the last question is, do you use a cell line? No, sorry, how do you establish the dose of the drugs that you use?
Yeah. So to establish the dose of drugs, we went to some lengths to do that. And we actually started this work using Francisella novacida, frankly, because our cell cultures were down, our Anaplasma cell cultures. And so with Francisella novacida, it also, invades these tick cells the same as marginale, and you have the, ability to just culture it on a plate. But when we did that, we went through and we did dose response curves to show at what, to see that we got a dose response as we increased the dose with the tick cells. And then we also went in and we did viability counts using trypan blue. So we know that we have chosen doses that are not lethal to the cells. The cells remain healthy, and that we have seen in a different system a dose response to give us some indication that these do what we think they do. Because again, nobody works with these things in tick cells. It's all done in mammalian cells.
Mm-hmm. And I have used one personal question. Well, let me see if there is. Yeah, I have one personal question. What do you think of the variability of these surface proteins in terms of vaccine for Anaplasma marginale?
Oh, well, I think so. I think ultimately we need to target the adhesins, and we also need to target other surface components to get a robust vaccine. I think the adhesins will allow us to target highly conserved areas. So for example, OMP-A, it's highly conserved across, strains from all over the world. There's very few changes in that. We have immunized and challenged with that protein, a recombinant protein. We got lousy protection with that. We used two proteins, another one that's maybe an adhesin, but it hasn't been shown. And when we went back and did some epitope mapping, we discovered that the immune system, the antibody, we just measure antibodies, antibodies were not directed against that actual binding domain. And, you know, in hindsight, that makes really good sense, in that those binding domains probably should not be immunogenic if the bacteria wants to survive. And so I think to leverage adhesins, which my suspicion is that they're going to be more conserved than not, we need to find those binding domains and force an immunological response against them. Whether or not you could then, whether or not that would be sufficient protection to protect against all strains, I think is still an open question. But that's, you know, I think one of the advantages of going down this, maybe we can find subdominant or poorly immunogenic, functionally essential genes to target.
Mm-hmm.
But it's a little bit waffly because frankly, I don't know.
All right, we'll see probably in some years later.
Let's hope.
Well, thank you very much, Susan. It was nice to see you again.
All right, you too, and thanks for the invitation, and wish I could see you all, but bye.
Bye-bye. Vamos a continuar con la siguiente ponencia, pero ahora quiero tomar tiempo para aprovechar para ver el video de nuestros patrocinadores de Elanco.
Elanco acaba de expandir sus horizontes. Ahora, toda la línea de productos de salud animal de Bayer, reconocida mundialmente por su alta tecnología y calidad, hace parte de nuestro portafolio y llevará a nuestra marca por nuevos caminos. Es así, expandiendo su participación y seguimiento, que Elanco consolida su liderazgo e innovación en la prevención y tratamiento de animales de producción y animales domésticos. Bienvenido al movimiento Elanco de crecimiento saludable de las relaciones de negocios, pero lo más importante, de la salud y el bienestar animal. Elanco, porque crecer es saludable.
All right, the last speaker of this second day of our international symposium on tick and tick-borne diseases is Dr. Guilherme Klafke. Hello, Guilherme. I think you're...
Hi, Juan. How are you?
Good. Good to see you.
Good to see you too.
Can you go ahead and transmit your presentation?
Yeah.
Mm-hmm. All right.
Right.
Well, I think we're ready. I'm going to give a little presentation of you, Guilherme. Dr. Guilherme is a researcher at the Animal Health Research Center, Department of Agricultural Research in Rio Grande do Sul State, Department of Agriculture in El Dorado do Sul in Brazil. Dr. Klafke holds a degree in veterinary medicine. He got it in 2001, and a master's degree in 2006, and a Doctor of Science degree in 2011, all of this from the University of Sao Paulo. He has experience in animal health with an emphasis on parasitic diseases, such as control of bovine ticks, diagnosis, and evolution of acaricide resistance, and molecular biology of parasites. So he will be presenting his research on acaricide resistance. Thank you, Guilherme. Thank you for accepting our invitation.
All right. Thank you, Juan. I want to thank you for the invitation as well as for the opportunity to talk this afternoon. I'm bringing here some of the development that I've been doing, not only me, but a lot of people that we are working together to try to give more light into the acaricide resistance problem that is one of the biggest issues for the cattle tick control in cattle, and tick control in cattle, but also in other species of animals. So, as I said, I work for the Department of Agriculture, the State Department of Agriculture in the Rio Grande do Sul state. We are in the South Brazil. We are close to the border with Argentina and Uruguay. So there's a kind of different climate and conditions here from the rest of the country. So we're more in a subtropical area. So there's some peculiarities here in terms of cattle tick producing. It's different from the other places in the country. So I work for the Institute of Veterinary Research here, working at the parasitology lab. Just to start, just to bring into the conversation the amazing diversity of ticks. They evolved across hundreds of millions of years, parasitizing different kinds of animals, terrestrial animals. So it's a very adaptable animal, parasite, that can basically survive for at least 3 mass extinction events in the history of the planet. So we can from that we can take that he's a very persistent and very hard to eliminate species of parasite, right? It's very adaptable. It can survive in different circumstances. It's an amazing animal. But in this presentation, we're going to talk more about the Hypcephalus microplus tick, which is part of a complex of species, which Hypcephalus microplus, Hypcephalus annulatus, and Hypcephalus australis, they are together in what we call the cattle ticks, right? They are one-host ticks. They prefer cattle for feeding, and they are present in most of the. In the case of the microplus ticks, they are present in most of the subtropical and tropical areas of the planet, and you can virtually find this tick in any subtropical and tropical area where you have cattle. Of course, this tick is displacing some indigenous ticks in Africa, like the Hypcephalus decoloratus, and it's invading the sub-Saharan Africa in a very aggressive manner, and it's a major problem for cattle producing in many countries because of this vectoring of Babesia and Anaplasma. Dr. Joe Bush already and Dr. Masato talked a lot about that. This is our model for study. It can harm cattle in different ways, like because of the blood feeding and transmission of diseases, and also injection of the salivary. The saliva is full of compounds that can be very toxic to the cattle, and in terms of like when you have like high burdens of cattle, it can create a problem for the production, right? So because of that, we need to control the ticks. And the thing is, for at least 100 years, we've been using synthetic acaricides to try to eliminate this tick from our herds. We have, in the market today, we have 6 classes of acaricides, like synthetic chemical acaricides, like organophosphates, pyrethroids, amitraz, macrocyclic lactones, fipronil, and fluozuron. in different forms of application, like dipping vats, pour-on formulations, injectable, or spraying. So we have the tools to control those ticks, right? But what we see along the time is that the ticks have developed resistance steadily to all the classes available we have in the market, right? So since the 1960s, of course, I'm not counting here with the cyclodians and arsenicals, but from the chemical classes that we have available, we have resistance being steadily developed across the time. And interestingly is that Brazil is, from the modern drugs, we'll say, it's always been the first one to detect resistance worldwide because of the high number of cattle and the perfect conditions and the high use of parasites. In the arthropod pesticide resistance database from the International Committee on Pesticide Resistance, Rhipicephalus microplus has 562 resistance reports in this database to 50 different active ingredients. So it's a very adaptable pest. In the Americas, we have virtually resistance in all the countries where this tick is endemic. In the United States, of course, is only in the Dr. Joe Bush presented very well the information about the presence of ticks in southern Texas. So that's the only place we have resistance there. And but in some other countries like Brazil, Argentina, Mexico, of course, Colombia, and Uruguay, the resistance is very problematic because we already have resistance to 5 or 6 classes of parasites already detected. So it's been very hard to control those ticks in these conditions. And we also see resistance in the Rhipicephalus sanguineus group developing in these countries like in the United States, Mexico, Panama, and Brazil. And we also detected resistance to ticks of the Amblyomma cagenense group in Mexico and also in Brazil. So it's all those ticks are very important in terms of animal health and, of course, human health in case of Amblyomma and Rhipicephalus sanguineus. It's becoming very a point of concern. Well, in Brazil, the resistance to 6 classes is present in the major cattle producing areas of the country. Like the states that are here marked in red are very important for cattle producing. Here, Rio Grande do Sul is where we are now. It's right here. And in other states as well. So it's a big problem for Brazil. So with that, what we need to do, the most important thing to do in terms of parasite resistance is to do the diagnosis of its occurrence, right? So that's important to confirm if a treatment failure is due to a parasite resistance. A lot of times we have failures of control, and we also have misuse of the parasite, like wrong way of diluting the parasite of our low doses that are not sufficient to kill the tick. So we need to separate what is a bad use of the parasite or wrong use of the parasite from resistance. Parasite resistance diagnosis is important for the decision making in changing or rotating the parasites. And it's very important for to monitor for early development of resistance to a new parasite, right? When you start using a parasite, and you keep monitoring to see when you're going to start to develop resistance. We have available like bioassays or toxicological tests that are classically used to detect this resistance. which are the adult immersion tests that you can do with the commercial formulations of the acaricides. You can use the larval immersion test or the larval packet test, so we can detect resistance to the major classes of acaricides. To bring an example of what's going on here in our state is we, here at the laboratory, we receive ticks from different branches across the state to perform the bioassays and detect resistance to help the producers in choosing or in the management of the tick control. So here I'm presenting tests that, like the scenario of acaricide resistance in Rio Grande do Sul from 2015 to 2019. We have results from 619 tick samples from 108 different locations or municipalities in testing the different pesticides. What we've been seeing, at least for the last 5 or 6 years, is that pyrethroids, we have a very prevalent, the high frequency of resistant populations, right? So each of these bars represent the frequency of resistant populations to a different chemical to a different in a different year. So we are seeing more than 90% of populations that are resistant to pyrethroids in the state. Is not different from what I believe is not different from what is going on in Mexico, for example, and that's what we saw on the Joe Bush presentation, showing the genotypes associated with resistance to pyrethroids. For amitraz, we have about 80% resistant populations. Ivermectin, 70%, fipronil, 70%. The organophosphates, the frequency of resistant populations is lower. And the association of the mixtures, right, the mezclas of acaricides, they're still working for some of the formulations, like the cypermethrin chlorpyrifosfenone, which is another organophosphate at a high concentration of organophosphate. It's being quite okay to control the ticks here. But we must say that this dependence on the acaricides is the major problem, because we don't have any alternatives to control those ticks, right? So what happened is that we have now the multiple acaricide resistant populations. So this is a graph that shows to how many acaricides a single population is resistant in the state. So we compile the data from all these populations that we tested, the ones that we have tested for 7 different chemicals. And what we see is that 62% of those populations are resistant to 4, 5, 6, or 7 different acaricides. So those are the super ticks here in red. We can call it the super ticks, right? Because you virtually cannot control those ticks with acaricides. And what is happening is that this breaks all the sustainability of the cattle tick producing here. And there's reports of producers that they want to sell cattle and just start growing eucalyptus or soybean, because it's easier than deal with the cattle and the cattle tick problem, and of course the tick fever problem. So it's a big issue. And then we have the super ticks. The super ticks, we reported the first strain of tick resistance to all the chemical classes back in 2014, including fluazuron resistance. And we've been seeing this trend in different states of the country, but also in other countries in the Americas, like Colombia, Ecuador, Uruguay, Argentina, and in other countries like India, places in Africa, and of course in Mexico. So that's a trend, and what we can say is that the cattle centric way of controlling ticks must be reviewed. On the way that we raise cattle, or the system that we raise cattle, must be reviewed, because it's been showing that it's not working. Only the use of a parasite is not going to solve the problem. It's cattle tick control is a complex system, and we need to change find a new way to look at the problem, instead of just, well, which is the pesticide to use. So there's a lot of efforts in developing vaccines using biological control, using botanical pesticides, for example, to try to control those ticks. But if we don't change the system, which is of course it's a very complex discussion, of course, because we have the need for animal protein, we have all that sociological and economically important issues involved. But the way that we produce cattle in the world right now, in terms of industry, it must be reviewed, because it's not going to work. Well, the evolution of pesticide resistance and multiple resistance is also going through the other species of ticks, like Rhipicephalus sanguineus, and other Rhipicephalus ticks in Africa, for example, Hyaloma in Asia, and Amblyoma in Mexico. So we're going to face this problem in other species. We see this in mosquitoes, we see this in bedbugs, we see this in crop pests, right? So where do we go from here? So there's some needs that we need to work with, like do the epidemiological studies to establish risk factors in hot zones for multiple resistance to parasites, block the dispersion. The data that Joe Bush brought this morning about the panmixia of ticks that occurs in Mexico, and it happens here in Brazil as well. I don't believe we have any population structure of ticks in the state or in the states close by, but we need to block the dispersion of these multiple resistant ticks, and for that we need rapid detection. I think most of the people here are familiar with the bioassays and how we do that, but I'm going to bring that back later, how long it takes to take the result of the bioassay to see if the population is multiple resistance. So we need rapid detection, we need biosecurity measures, we need new molecules. But with a new molecule coming in to control those multiple resistant ticks, we need rational use of that new molecule. And we need to, and everybody needs to know that a new molecule is going to face the same fate of the other drugs that were available. We're going to have resistance in the future for any kind of new molecule that comes in to control the cattle ticks. But if we keep using acaricides like we used before, we're going to have a short time to count on the new molecule to control those multiple resistant ticks. And of course, we need integrated tick management, like do the rational use of the parasites, vaccines, botanical pesticides. We have biological control. We have host resistance. So we need to use all these tools so we can lower the pressure on the acaricide resistance selection by using synthetic chemical pesticides. And we need to really engage the producers and the society on these. So going back to the subject of the presentation, Joe this morning talked a little bit of the molecular tools that we can use to help on the tick management, right? So I'm going to talk a little bit more here about the development of those markers so we can detect resistance faster and in a way that we can help with the tick control. By comparing toxicological and molecular diagnostics, we can say that bioassays, anyone here that has experience of bioassays know that we need to collect those ticks in the field. We need to put those ticks in an incubator. They have to lay the eggs. Those eggs needs to hatch, and then we can run the bioassay and count the ticks and analyze the data. And that takes, because of the biology of the tick, it takes at least 6 weeks to be ready. So we know if a population is resistant or not to a given chemical. By the other side, the molecular detection helps us to get this information early. We have information available now, very robust information about the presence of mutations in the genome of the tick and associated with resistance to some chemical classes. So by combining this information, we can be very sure if a population is resistant to a given chemical by analyzing the genetic data of those ticks. So, and another advantage of using the biological or the molecular markers is that for the bioassays, we need to collect fully engorged females in a good number in the field, so we can have the eggs and then run the bioassay with the adult ticks, or running the bioassay with the larval stages. But with the molecular tools, we can go to the field, and it's not and it's frequent that we go to the field and gather the cattle and go to collect ticks, and you don't find too many engorged female ticks depending on the time of the day. So we need to collect any tick. You could collect any tick, like nympho stages or immature stages, or males or semi-engorged females. They're not useful for the bioassay, but we can still get genetic data from those ticks and get information even if you don't have engorged female ticks. So just to give an example of the time to obtain the results and a comparison of the bioassays and the molecular tools, I'm going to bring the example of the tick strain that was collected from Puerto Rico in a dairy ranch. They are doing a vaccination operation there, and they found ticks in cattle. And they sent the ticks to the USDA ARS Cattle Fever Tick Research Lab that I was working there for the past 4 years. We received those ticks there, right? Sorry, was a problem here. And in those ticks, we have developed a high-resolution melt analysis for detection of SNPs associated with pyrethroid resistance. There's a good review that was published last year about the mutations associated with resistance to pyrethroids in the cattle ticks. So we use this information to develop an AGRM, qPCR AGRM, to detect resistance to the cattle ticks, to pyrethroid resistance. So what happens is that you make a real-time PCR, and in the end you run a high-resolution melting analysis of those fragments. And with that, you can separate by the format or by the shape of the curves of the melting curves, you can separate the genotypes of the ticks in homozygous, wild type, resistant, or heterozygous, right? And you can compare those curves with standards, and you can say a tick has one or the other genotype. This is the KDR mutation. This is a super KDR mutation here. So on November 7th, we had the collections of the ticks in the field: 20 fully engorged females, 19 semi-engorged females. The fully engorged females, when they come to and they arrived to the lab on the 10th of November, 3 days later, right? We put those ticks on the incubator to run the bioassays, and the other ticks, we did the DNA extractions and we ran the HRM for the pyrethroid resistance. 7 days after the collection, we already knew that we had genotypes of associated with pyrethroid resistance in that population. So we could say that that population was possibly resistant to pyrethroids. 49 days later, after the collection, on December, we got the results from the bioassay confirming that that population, and this is the concentration of different concentrations of the chemicals that we tested, that strain, showing that a high level of resistance to pyrethroid. This is the mean mortality of ticks to the given concentration. Those are discriminating doses of resistance. So showing that that population was highly resistant to permethrin. But we already knew that 40 days before that by using the molecular tools, which is impressive. So we can use this data. What we need is to develop those markers for the other drugs as well, so we can have a better resolution of the resistance status of populations. Regarding the sample type, Joe talked a bit about the quarantine zone in the Texas and Mexico border. In the USDA APHIS has cattle inspection facilities on the United States side of the border. So every live cattle that is imported into the United States from Mexico is inspected. There's a picture of a inspector here looking for ticks in the cattle. And when those cattle are, when they find ticks in this cattle, they send the ticks to the lab so we can extract the DNA and do the surveillance, what's going on. And most of the time, those are nymphs or semi-engorged female or non-engorged females, males, or even dead females. And usually they found 1 or 2 ticks in a cattle only. So we can't actually run any bioassay with that. So what we did, we just extracted the DNA and we ran the HRM of those ticks. And we were able to say that those ticks were resistant to pyrethroids, right? So that's a way of tracking the resistance without using live ticks and running bioassays. So it's a very powerful tool. And what is interesting is that these markers can be adapted to other species of ticks as well, like Rhipicephalus sanguineus, Annulatus, Appendiculatus, Decololatus, because those mutations are conserved across the species, right? So we have been working with other species of ticks, trying to expand the possibility of molecular detection of resistance, especially for Rhipicephalus sanguineus, where we described a mutation in the sodium channel, in the domain 3 of the sodium channel, that are, and this mutation was associated with pyrethroid resistance, and curiously is on the same nucleotide or the same locus that we find resistant in Rhipicephalus microplex ticks. So we've been working a little with other strains of Rhipicephalus sanguineus to try to find other mutations. We also worked with Rhipicephalus annulatus. We found a population in an outbreak in Texas that was resistant, slightly resistant to permethrin. Here's the bioassay data showing a small number of surviving ticks in this population, showing that it was resistant. We ran the AGRM, but we couldn't find any mutant in this population, so maybe there's another mechanism of resistance. But we used the same technique in ticks from Egypt that were resistant to deltamethrin, and we were able to find the mutation associated with resistance to pyrethroids in Hypcephalus micropus. So it's a very powerful tool, and we can adapt that to other species. We started a project with the Amblyomma mixtum from Mexico with a collaboration of Dr. Barradas Piña from Veracruz, and we had access to ticks from Veracruz and Oaxaca from Mexico, and we ran bioassays on those ticks, and we found resistant to pyrethroids. So what we did, we designed primers based on the Amblyomma caginense sodium channel, and we were able to run an AGRM on those ticks, and we found some haplotypes that were associated here in red, haplotypes associated with the resistant populations. Unfortunately, this project was running when the COVID pandemics came, and we weren't able to sequence those genotypes to see if the mutations are possibly associated with resistance. But those ticks are in the DNA is in the freezer waiting for some work back on those maybe soon, but showing that it's possible to adapt those techniques to other species. We also have the molecular, so trying to increase the number of markers that we can use to detect the ticks resistance to the other chemicals. We worked a little bit with amitraz resistance. So in 2007, there was this description of mutations associated with amitraz resistance in the octopamine receptor gene. That was later investigated in Africa, in South Africa, and then later in India, showing that those mutations could be associated with resistance to amitraz. And we have ran an AGRM on some samples in the USDA that were resistant to amitraz. Here's Santa Luisa strain, El Zamora, and Yucatan. Santa Luisa has origin in Brazil, El Zamora in Tamaulipas, Mexico, and Yucatan from Yucatan state in Mexico. And we ran the AGRM on those, and we found the mutations associated, like previously associated with resistance, right, on Santa Luisa strain. But we also found new haplotypes here on El Zamora and Yucatan that we didn't know what it was. Also, we had access to ticks collected from New Guy, the same ones that Joe was talking about, and we ran the AGRM on those, and we've also found mutations associated with amitraz resistance, like these ones that were found in Santa Luisa, but also the same ticks, the same haplotypes that we found in other Mexican ticks, showing that most likely these populations here had an incursion from Mexico in New Guy or in this area, showing that this is an area of high movement of ticks, right? Because there's no use of amitraz in the United States, at least legal known use of amitraz. And we tried to sequence those haplotypes, but we never found any mutations associated, which is kind of weird because most likely this gene has multiple copies, and we are not able to find these mutations on the sequencing. But maybe if we go through like deep sequencing, we can try to find these haplotypes in other copies of these genes. So there's information about those different haplotypes going on. In Africa, in South Africa, they found that there's a lot of mutations that are associated with resistance. Sometimes they appear, sometimes they don't. And to bring another light to that is that we, here in Brazil, we took a resistance strain and we run bioassays using and synergists like the PBO, which is a synergist that disrupts P450s that mediate resistance with, and we found that PBO was responsible in reducing the LC50 or increasing the toxicity of amitraz in that population, and showing like a synergism ratio that was significant, showing that besides mutations in the octopamine receptor gene, we also can have metabolic resistance associated with amitraz. So it's a very complex and rich, interesting model in studying resistance, because there's has multiple mechanisms involved. And we also have the information about the beta-adrenergic like octopamine receptor and amitraz resistance, as was shown in Australia by a group there, showing new mutations associated with amitraz resistance, and another group in Japan showing that the region where the amitraz binds to the octopamine, beta-adrenergic octopamine receptor, is important, that a mutation there also blocks the binding of amitraz. So it's another aspect of amitraz resistance in the cow tick. Was interesting that we found these mutations that were found in Australia in the Santa Luisa strain that is resistant, but we never found these mutations in the other two. But we also found a new SNP on this position on the Santa Luisa strain. And we had ticks from Santa Luisa strain that had mutations of both octopamine receptor and beta-adrenergic octopamine receptor. So bringing more information about this. So, and we also found these mutations in ticks from Brazil and in ticks from Australia. So apparently this transmembrane region here is very important for amitraz resistance. And we also look into including those in resistant markers to help on the amitraz resistance detection. We've been working also with fipronil resistance. I'm just going a little faster here because we're going to the end. So a group in Australia found mutations SNPing the ticks that were resistant to dieldrin, which does act on the same site as fipronil. We showed here that in Brazil that we had cross resistance between fipronil and cyclodienes, showing that possibly the mechanism of resistance is the same, and we were able to find mutations in ticks that were resistant to fipronil here. What is interesting is that here ticks from Americas, I mean Brazil, Uruguay, and also Mexico, they have mutations. They are different from the ones in Australia. So, which is interesting in terms of evolution. They might have developed different mutations associated with the resistance, or also we can have multiple copies of those genes that have different mutations. So, and we were able to develop molecular to detect those mutations here. We have published that with a group in Uruguay that we can use to screen for fipronil resistance as well. So with that, taking all the acaricide resistance mechanisms that were already described for Hypcephalus microplus, we have this table here. We are able to detect the target site alterations with PCR. So we can use those molecular tools to detect. But On the other side, we don't have methods yet to detect resistance mediated by detoxification mechanisms. So Joe mentioned it about the project we have about the transcriptome of resistant strains. We're trying to understand how much the detoxification mechanisms or genes or markers can be found that we can use as tools to detect resistance and help on the management of the tick. With that, I think it's time for the questions. I would like to acknowledge the funding obtained for different agencies here in Brazil and the United States. And I would like to thank you. I can take any questions. Thank you.
Thank you, Guilherme. There are some questions here in the chat. Dr. Carlos Vega says, can a molecular assay be made quantitative? How do you plan to make a molecular assay quantitative?
Well, it can be quantitative if you take like a population of like test, I don't know, 20, 30 ticks, and you can calculate the frequency of the mutant alleles and say, well, with this information, you can say, well, this is a highly resistant strain or low resistant strain. But sometimes you just have one tick, right? Or just a few ticks, and sorry, it can't be quantitative in this condition. It depends on how many ticks you're gonna have access in genotype. Yeah.
Okay. The second question is, are molecular assays more expensive than bioassays?
Until now, yes. But we've been developing, we are working now actively in a LAMP detection method. So the loop mediated assay. So we believe that with the cheaper molecular method, it can be comparable if you take the time. I mean, if you put the time in the time to get the results in the calculation, I think it's worthy to have access to the information. Like, if we want to get to the point that we can have like a rapid test to use in the field. So you go to the range and you collect ticks and you do a crude extract of DNA and just put in a reaction, and you know there in the pans that you can use, or you can choose any kind of pesticide. Of course, we're kind of far from there, but I believe we can get to the point that that's very commercially viable to use the tubes.
All right. Dr. Dora Romero Salas says, excellent conference, especially because the problems that are occurring in the cattle ranch of the tropics of Mexico, and especially in Veracruz.
Yes.
Greetings to you.
Veracruz is it's a very important area, right? And we got, it's that part of the study we did with the Amblyomma myxoma, we got ticks from Veracruz, and we were able to, I didn't talk this publication, but we were able to test mycoplas and Amblyomma ticks from the same location. And it's very interesting that when you find resistance in the mycoplas, you also find resistance in the Amblyomma ticks, because they affect the same cattle, right?
Okay. All right. Well, Guilherme, I really appreciate you to participate in our symposium. It's the information that you provided is very important, because as the same as in Brazil and other Latin American countries and Asian and African countries, the problem of acaricide resistant doesn't have like we don't see a light. I don't know when it is going to finish, or if ever is going to finish.
Yeah, I don't think it's very hard to respond that question, Juan.
Yeah.
Sure.
So thank you very much. And I think that with that, we are finishing the talks for today. Thank you, Guilherme.
Thank you, Juan. See you.
Bye-bye.
Bye. Good to see you.
Con esto damos por terminadas las conferencias de hoy. Les agradecemos su asistencia y antes de pasar con el doctor Eleazar, vamos a ver el video de nuestro patrocinador, el ANCO. Muchísimas gracias a todos. Nos vemos mañana.
El ANCO acaba de expandir sus horizontes. Ahora, toda la línea de productos de salud animal de Bayer, reconocida mundialmente por su alta tecnología y calidad, hace parte de nuestro portafolio y llevará a nuestra marca por nuevos caminos. Es así, expandiendo su participación y seguimiento, que el ANCO consolida su liderazgo e innovación en la prevención y tratamiento de animales de producción y animales domésticos. Bienvenido al movimiento del ANCO de crecimiento saludable de las relaciones de negocios, pero lo más importante, de la salud y el bienestar animal. El ANCO, porque crecer es saludable.
Muchas gracias, doctor Mosqueda. Agradecemos la participación de todos nuestros ponentes que nos acompañaron el día de hoy y los invitamos para que el día de mañana nos acompañen en el último día de la transmisión del cuarto Simposio Internacional sobre Garrapatas y las Enfermedades que Transmiten. El día de mañana todas las pláticas van a ser en español y podrán seguirnos a través de nuestros dos canales que son Facebook y YouTube. Y cabe señalar que el día de mañana, pues al igual que los días anteriores, las pláticas van a estar dadas por investigadores y expertos con un enfoque de investigación aplicada y experiencia en el campo. Entonces, los invitamos para que el día de mañana nos acompañen a continuar en este último día de actividades y continuar recalcándoles la dinámica de lo que es el simposio. Cada ponencia tiene una liga en la cual ustedes pueden registrar su asistencia y tendrán solamente 10 minutos para poder hacer el registro. A partir del día de mañana nosotros vamos a contabilizar su porcentaje de participación y basado en el porcentaje les haremos llegar sus constancias. Pues muchas gracias a nuestro patrocinador el ANCO y a todas las instituciones que estamos organizando este evento. Gracias a los participantes que nos acompañaron el día de hoy y nos vemos el día de mañana. Buenas tardes y muchas gracias.
El ANCO acaba de expandir sus horizontes. Ahora, toda la línea de productos de salud animal de Bayer, reconocida mundialmente por su alta tecnología y calidad, hace parte de nuestro portafolio y llevará a nuestra marca por nuevos caminos. Es así, expandiendo su participación y seguimiento, que el ANCO consolida su liderazgo e innovación en la prevención y tratamiento de animales de producción y animales domésticos. Bienvenido al movimiento del ANCO de crecimiento saludable de las relaciones de negocios, pero lo más importante, de la salud y el bienestar animal. El ANCO, porque crecer es saludable.